Okay, hello. Thank you so much for attending mga ma'am at sir. We are going to start admitting our participants now. Ayan. Mm. Uh, Hello, everyone. Thank you so much for joining us on this really nice Friday afternoon for Intersections. How scientists use art to explore the world. Talagang pinaghandaan po namin ito para sa inyong lahat, and we're sure it will be a very interesting afternoon for everyone here. Gusto lang po namin ipaalala po before we start that we would like to remind all the participants of the house rules that we should follow through this webinar. No? Unang-una po sa lahat, sana ma-mute natin yung sarili natin. Audio should be muted when the speakers are talking. Unless, of course, at the question and answer session or if you're asked to speak. No? So wag po tayong mag-alala. Meron po tayong opportunity to air questions, to ask questions towards the end. We will provide you enough time for that. So sana po, for everyone's enjoyment, let's mute ourselves para mas napapakinggan po natin mga speakers natin. Pangalawa, we would like, if possible, for you to identify yourselves na kung pwede po natin baguhin yung pangalan natin sa Zoom, yung full name ng participants at yung affiliation, sana po mailagay po natin. Makakatulong din po sa atin para mas ma-address namin po ang mga concerns ninyo. We will be able, and also it would help us get to know you a little better. So makikita niyo po right now, nasa ilalim po ng screen ko ng screen ko ang buong pangalan. Ako po si TJ Dimacali, taga UP Diliman College of Science, UPDCS. Kasama din po namin si Uni, kasamahan ko rin po sa UPDCS. So we're looking forward to hearing from you later on. Pero yun, after po magsalita ang ating mga speakers. Finally, yung Q&As, magkakaroon po tayo after magkakaroon po tayo towards the end. We would like to ask you to submit your questions directly via direct message sa amin po, either sa akin, si TJ po ulit, or si Uni, so that we can look over them. Tapos panghuli, additionally, we would like, at the end of the entire event, we will have an evaluation form that will be sent out online. This will serve as attendance for everyone, and this will also be the basis for certificate requests. So dun po sa atin na nag-aabang po ng certificates, we will definitely be providing them, but we will ask you to provide information na ilagay po sa evaluation form at the end of the event. Meron po nagsabi dito sa chat, I saw it just now. Hello, Lorenzo and Samantha. If you have trouble adjusting your names, that's okay. We'll see if we can change settings on our end. Pero kung hindi niyo mabago, that's okay. Siguro what we can do is during the live Q&A session, if you decide to speak, we can feel free to introduce yourselves to us, to the speakers later on. May tanong din po tayo dito, can I share the meeting with other people who might be interested to listen in? The Zoom meeting po is closed to the participants. We're very happy to say that there was such a large demand for the Zoom meeting. I'm sorry po, but we cannot share the Zoom meeting outside. However, pwede nyo pong sabihin po sa mga kaibigan nyo na pwede po silang manood through the Facebook live event. Pumunta lang po sila sa Facebook page po namin. That's facebook.com/UPD-Science. Ulitin ko po, facebook.com/UPD, University of the Philippines Diliman Science, UPD Science. Nandun na po yung live stream. They can watch the entire proceedings there. Tapos, we will also try to field questions from there as well later on. Po. And dun po sa mga hindi makaka-attend, meron din po kasi nagtatanong, we will also provide the full recording, which will also provide on Facebook and our YouTube channels after the event na po. Yun lang, hindi po namin masisigurado kung kailan po namin ma-upload. Hindi man po yun agad-agad later this afternoon. But we'll try our best to upload it as soon as possible para sa mga hindi naka-attend ng event. Gayon pa man yun, once again, it's really good to have you here this afternoon. At syempre, mas masaya naman po, no? Nandito tayong lahat ngayon, nagsasalo-salo. Mas lively. Hopefully, these talks will be a very lively and an engaging opportunity for us to learn more. Hindi lang about science, but also on how art helps with science and how science actually also benefits art, no? So, with that, ulitin ko lang po ulit yung ating house rules. Yun, please stay muted during the talks so that everyone can listen properly. Identify yourselves when you are asked to. Also in the Q&A. The Q&A itself, we will provide a time for you to speak during the Q&A or to write your questions. You can direct your questions to me or to my colleague and co-host, si Uni. And at the very end, for those who are interested in the certificates, we will provide an evaluation form. We will send a link through the chat room so that you can avail of your certificates. Ayun, maraming salamat po.
Ayun po, no? Magandang hapon po sa ating lahat. Good afternoon. I am Uni.
At ako naman si TJ.
And we are your hosts and moderators for our afternoon webinar today. We have participants via Zoom and our Facebook live stream. Welcome po everyone sa ating
Welcome
webinar.
po sa ating lahat. Yon. Thank you so much for spending your time with us this afternoon para dito sa Intersections, How Scientists Use Art to Explore the World, presented by the UP Diliman College of Science.
Ayun po. So para alam lang po ng lahat, every February is the National Arts Month. We hold the National Arts Month to celebrate artistic excellence and pay tribute to the uniqueness and diversity of Filipino heritage and culture. And para makasali tayo sa ating celebration na ito, the UPD College of Science organized this webinar featuring UP CS scientists who will share how their work was enriched through their love and pursuit of the arts.
Tama yun, no? So, dito natin makikita na yung mga scientists, hindi lang tayo sa lab lang, no? Hindi lang nasa labs lang scientists. We also express ourselves through art. And art also is an important part of our work. These scientists who you'll meet this afternoon are not just involved in the field of science, but also are immersed in the world of art itself. Tamang-tama para sa National Arts Month, no? Although most people usually view science and art as separate disciplines. Kasi syempre, nung nag-aaral tayo, di ba, magkaibang subject yun. But our guest scientists today don't just believe, but they know otherwise. Na isa sa buhay nila yung intersection nga ng art at science. They draw inspiration for their work and for their art from both art and science as they explore the world.
And I'm sure all of us here are excited to learn more about them, their passion for science and the arts, and how their work was enriched through their love and pursuit sa arts. So without further ado, let us formally open this forum with a message from the Associate Dean of the College of Science, Dr. Lilian Jennifer Rodriguez.
Good afternoon po sa lahat. Maraming salamat po sa pagsama sa amin ngayong hapon dito sa Intersections, kahit na holiday ngayon. So gusto po namin na malaman ng lahat kung gaano ka-importante ang arts sa science and yung science din sa arts. So ngayong hapon po, in celebration of National Arts Month, nag-imbita po kami ng 4 na siyentista mula sa aming kolehiyo. Itong 4 na tao na maririnig niyo po ngayong hapon ay mga doktor ng agham. So mga doctor scientists po sila. that are also artists or art lovers in their own right. So ipapakita at ipaparinig po nila sa inyo kung paano naging istorya nila sa buhay at paano nakatulong ang art sa kanilang buhay bilang scientists. So again, maraming salamat po, and let's all enjoy the talks. So happy National Arts Month.
Happy National Arts Month. Maraming salamat po, Doc Lilian. Thank you so much for welcoming everyone and for kicking off the event. So ngayon, I'm sure excited na kayo sa mga speakers natin. So umpisa na kaagad natin. No? Let's start with our first guest. He is a marine biologist from the Institute of Environmental Science and Meteorology. He studies intertidal and marine invasive species. And he also writes prose and poetry on natural history. Palakpakan po natin. Let's all please welcome Dr. Benjamin Vallejo Jr.
Okay, magandang hapon. Salamat sa pag-imbita dito sa ating discussion for Arts Month, no? So ako ba mag-share ng screen? Jane? Sige, ako ba? Sige,
Yes. Yes.
Sige. So mag-share lang po ako ng screen. Okay, ito na yun.
Sige po.
Okay, so yung aking parang reflection na rin to, presentation, what does a starfish know, no? Ang tanong niyo, bakit pa kailangan alamin kung anong nalalaman ng isang sea star or starfish? My presentation is essentially about writing. Okay, now one of the things that marine biologists traditionally do is to have beer at the end of the field day. Kaya nga sabi nga dito, marine biology plus beer equals perfect day. Okay? So I think maganda naman ang panahon sa labas, no? This is actually a nice time to go to the beach, no? Unfortunately, holiday natin late na dineclare. So wala time, no plans to go to the beach. It's so hard to get things to go to the beach right now, no? So let's just look at the scenery na lang, no? Now, okay, so beer plus marine biology equals perfect day. Okay, this was not lost on even your starfish. Now, I wrote a little bit of a ditty for this day, no. So all I know is that I had too much drink, life in all its wonder in my pint. Voices loud and profligate, Steinbeck and Ricketts, what good men marine biologists are. Okay, ipagpatawad nyo kung medyo parang sexist because yung ating pag-uusapan ay mga manunulat na scientifico, no? Tatlo ang ating example, si John Steinbeck, Edward Ricketts, at si Rachel Carson, no? Well, hindi po lalaki si Rachel Carson, no? Pero nung panahong yon, nung early part of the 20th century, karamihan ng mga marine biologists ay lalaki. Ngayon, si John Steinbeck, Edward Ricketts, and Rachel Carson, malaki ang naiambag nila sa marine ecology as a modern discipline and also for writing and philosophy, no? So paano nila yan nagampanan, no? How were they able to do that, no? Because they were writers, no? John Steinbeck, probably you have heard of him. He's in the canon of literature, of American literature. Mag-canon, yun yung required reading sa GE program sa US. Hindi ko lang alam sa mga colleges natin kung required pa rin si Steinbeck, no? Ang kanyang famous works ay The Grapes of Wrath and East of Eden, no? Pero nagsulat siya ng isang literary reflection on his work collecting marine specimens of the Sea of Cortez in Mexico, together with the marine zoologist Edward Ricketts. So meron silang pagkakaibigan na matalik, no? Sumakay sila sa isang maliit na barko, yung fishing boat na Western Flyer. And hanggang ngayon, pinag-aaralan pa rin yung kanilang, ano yung ginawa nila, no? Yung kanilang literary work and also the scientific work that came out of one marine biological expedition. So when they came back, it was marine biology and marine science changed forever, and the two scientist writers changed also. So Steinbeck had to ask this question. Okay, there are answers to world questions that everyone must ask in the little animals of the tide pools. One thing we marine biologists do is to observe for a long, long time exactly what we're looking at in the ocean. No, sometimes inordinate amounts of time, especially if you're studying animal behavior. Exactly what Steinbeck did, no, in their work, no. And they collected a lot of specimens, and marine biologists today still collect a lot of specimens and put them into the museum. Although ngayon mas mahirap na ngayon, you have to get all sorts of permits, no? But still, it's important to collect specimens, no? For documenting marine biodiversity and environmental change. And you have to write something about it. It's not just collecting specimens, no? Even if you go on a boat like the Western Flyer. In fact, one thing in my bucket list is to find, if I do win in the lotto, I should be able to buy a boat and sail around the country, okay, collecting specimens. But of course, I have to get lucky, no, and win the lotto. So Steinbeck and Ricketts went around the coastal part of California and Mexico and collected specimens, which he published in a book, no, The Log from the Sea of Cortez, which is always been an inspiration of most marine biologists, no. Most marine biologists they start in their careers, they start by reading this, and also the zoological manuals that came with the Log of the Sea of Cortez. Now, why is it so important to marine science and also in literature? For one thing, we can probably see it in Steinbeck's scientific writing. Right? So he writes, no, the Mexican Sierra, a kind of fish, has 17, 15, 9 spines in the dorsal fin. If you are a graduate of the College of Fisheries in UP Visayas, right from the start of your freshman days, you know how to count the spines of the fish, no? So Steinbeck was a marine biology undergrad, but he did not finish his degree, decided to go into writing. So when he went back to marine biology, he was able to write it in science and also in prose. So he said that if the Sierra strike hard on the line so that our hands are burned, if the fish sounds and nearly escapes and finally comes in over the rail, his collar pulsing and his tail beating the air, a whole new relational external reality has come into being. Now, how can you say that it's just looking at the fish and counting its meristics? But there is more to that than that. You have to observe, no, and see how the fish really is. Okay? So sabi ni Steinbeck, a new relational external reality has come into being, and he put it in prose. No, the only way that we can actually write about ourselves and reality is actually to put it into words. Now, the words could be in poetry, in prose, or in song. Okay, that's why we're quite about singing, no? Because singing hits our hearts very hard. No? And Steinbeck continues, and here's my friend from the National Museum who's going out all over the country doing things that Steinbeck and Ricketts does for the country, no? Collecting. Si Joseph Comendador is National Museum. Okay, he helped me in my collecting before, so maybe we agreed on this. The lies that we tell about our duty and our purposes are the meaningless words of science and philosophy are walls that toppled before a bewildered little why. If you do science and art without asking questions, then it's totally without purpose or useless. So we need to answer certain questions if we're writing and also if we are doing science. And for the marine biologists, there are ways of looking at marine biodiversity. You can see them in the museum, dead and preserved, or you can look at them in their habitats, or you can observe them in an aquarium. Each and every way of doing so will give you a new reality on what the animal really is. Okay? If you see it dead, it will always be dead, no? But if you see it, it will be different if you see it in the ocean, in its habitat. No? So a lot of the material of Ricketts and Steinbeck and his assistant John Calvin were put in the museum in Monterey, California. This is one of the so-called holy west shrines for marine biologists, because a lot of the modern ideas now that we have about the environment and ecology came from that single expedition to collect all of these specimens from the Gulf of California in Mexico by two remarkable people, the zoologist Ricketts and the Nobel Prize winning writer John Steinbeck. Both of them took degrees in marine biology but never got a degree, for a lot of reasons. So they say that you have to look at tide pools and try to find out exactly what the animals are. Observe the relationships between the two. On the left side of this slide is from the Steinbeck Museum in Monterey, California. I borrowed it from their website. Is the tide pool in California. And on the right is what we collected from Mactan, the port of Mactan in Cebu. And so the sea squirts on the right and the sea anemones on the left, what are the relationships of these organisms to each other? Okay? These things you can never, and including the starfishes on them. No? This is something that you'll never get if you just read it in the textbooks or if you just look it up dead in a museum. And the way to make them come alive for other people, because not all people have the chance to go to the tide pool and look at these animals for a long, long time. Remember, some people have other things to do, no? Instead of just looking at these animals. One way to make them come alive is to put them in words. So look at the animals, as Steinbeck said, and put them into words. Now, one of the quite interesting chapters in the or passages in the log of the Sea of Cortez is about the Sally Lightfoot crab. We have this in the Philippines, no, but it's a different species. The one that you see here is from a natural history guide that's in the public domain, and this is the Sally Lightfoot crab's species, or in the scientific name is Graspus, okay, in Mexico. So the Mexican species, this is the one that Steinbeck saw, is more colorful than the species that we have here. We have a Graspus species in the Philippines, but it's more of a greenish color rather than bright red. Steinbeck writes a long passage about the behavior of this animal. It's so hard to catch. That's why they called it Lightfoot, because they jump from one rock to another, and it's almost impossible to catch them. So Steinbeck will put it into words with actually, if you read it, it's as if that you are there looking at the crab, no? Although the book doesn't have any figures, no? The figures are in another separate volume. So, and if you go to the beach and see your first Sally Lightfoot crab, one of the most interesting things that happened because of the Manila Bay cleanup is that the species has come back to Manila Bay. So if you're lucky enough, and I hope And if you're lucky enough and they stop the reclamation, then probably you can find a salilite foot crab near the CCP complex. Actually, I did see one. So we're talking about starfishes and what do they really feel? No? That's actually a rather philosophical question. We are just observing the starfishes, but we're not really starfishes. We know the structure of the starfish. It's an echinoderm. They're dermal plates. The circulatory system is, they have a water pump type of circulatory system. They have many tube feet. It's pneumatic. It's hydraulic. The most things amazing about marine biodiversity is there's a diversity in how animals move and how they can move. So sea stars, actually the better term is sea stars rather than starfish. They move through a hydraulic process. But of course, their movements can only be better contextualized if you look at the general ecology of the habitat where they are found. Now, in marine ecology, there is always an approach to systematize the components and the relationships of the of marine biodiversity in the environment. And that would provide a lot of material for writing prose. And that's what Steinbeck did. Okay? And people who still write natural history manuals actually try to put the scientific observations into prose. Leading to Steinbeck writing later on that there is something for everyone in the sea, incredible beauty for the artist, excitement and danger of exploration for the brave and restless, an open door for the ingenuity and inventiveness of the clever, the new world for the bored, for the hungry, an incalculable material wealth for the acquisitive, and all this in addition to the pure and clean wonder of increasing knowledge. This is the point about the arts, the humanities, and the sciences. They are ways of gaining knowledge about the world. Their approaches are rather different, but the impetus to do so is more or less the same. In a very specialized world that we have, we tend to split the sciences from the arts and the humanities. Sometimes we try to shoehorn these sciences to serve art. Okay? We try to explain the arts in a scientific way. In a lot of senses, that does injustice. Something that the cosmologist and Jesuit brother Consul Magno, who was the director of the Vatican Observatory, in a homily, in a mass, he said that he is a musician too, and also a physicist and cosmologist. But he says that my knowledge of the physics of sound does not lessen my love for the beauty of music. While physics cannot describe beauty, neither science can describe beauty at all. There's no scientific theory to describe beauty. Everything is approximation. Even the Greek ideal of the golden mean is an approximation. It's an abstraction for beauty, but does not approach beauty in its complete humanistic sense. And it's only art that can do that. Now, the sciences can provide one way of approaching beauty, okay, without removing the meaning of beauty in the world. Okay. Now, something that Steinbeck and Ricketts in their marine biological expedition to the Sea of Cortez in Mexico, they realized it. It's a sort of breaking through. You break through the existing preconceived ideas about science and art to reach a new understanding, only to find out later on that you went back to science. And your view of science in the scheme of things, of gaining knowledge, probably has changed. So Steinbeck received the Nobel Prize for Literature in 1962, and it was a challenge to writers. The ancient commission of the writer, according to him during his Nobel Prize acceptance speech, has not changed. He is charged with exposing the many grievous faults and failures, with dredging up to the light our dark and dangerous dreams for the purpose of improvement. And what does the writer do and the scientist at the same time? Maybe it's also to expose a lot about our faults as humans. If you're working on the environment, you can actually expose a lot of the stupid things that our species have done to the environment. We can do it in a scientific way. But the arts actually would dredge up all our hopes and hopes for improvement. Of course, we need science to improve, but the impetus to really commit to improvement may be found in the humanities, with the sciences, with it. So here we see Ricketts collecting on the tide pools, meditating at each and every time collecting, finding out the relationships between organisms in their environment and in their general ecological communities. Similarly, Rachel Carson, another marine biologist that worked on the other side of the ocean, the Atlantic side. You probably know more about Rachel Carson as the woman who inspired the environmentalist movement by writing Silent Spring. Okay. In a time when science was very sexist, she wasn't able to get her PhD simply because she was a woman. Okay. No committee could accept her. No graduate admissions committee accepted her because she was a woman. But she still persisted in her marine biological work and changed environmental science forever by linking the ocean, her knowledge of the ocean, and the effect of pollutants. The effect of pollutants. And so we know that environmental impacts are not just local, but global, thanks to the pioneering work of Rachel Carson. So her most famous book is Silent Spring. You can Google that. No? Change our view of the environment forever. Change our scientific theories for the environment forever. But her first book is actually the best. It's Under the Sea Wind, which describes her early career as a marine biologist. And just like trying to find out the answers to the sea star, okay? What the sea star feels. Rachel Carson writes in Under the Sea Wind, to get a feeling of what it is like to be a creature of the sea requires the active exercise of imagination and the temporary abandonment of many human concepts and human yardsticks. For example, the time measured by the clock or the calendar means nothing if you are a shorebird or a fish. You have to break through outside your human senses in order to know exactly how a fish reckons time. Now, Steinbeck and Ricketts came to the same conclusion. You need imagination and temporary abandonment of many human concepts to understand marine ecology and marine biology and marine science as a whole. And what is the best way to know the world with imagination? That's the arts and the humanities. So that's what I try to reflect on every time I go on a field trip. Here I'm seen collecting seaweed as part of a research on climate change. No? At an exposed reef in northern Luzon. No? Or taking pictures of intertidal fish. I wonder what the goby feels every time there's a high tide coming in. Something I try to reflect on. And so it could be spiritual, as what Steinbeck realized in the Log of the Sea of Cortez. And this is actually the most quoted of all the passages in the book. Generations of marine scientists and marine biologists quote this sometimes if they feel depressed. Okay, and let me read it to you. It is a strange thing that most of the feeling we call religious, most of the mystical outcrying, which is one of the most prized and used and desired reactions of our species, is really the understanding and attempt to say that man is related to the whole thing, related inextricably to all reality, known and unknowable. That all things are one thing, and that one thing is all things. Plankton, a shimmering phosphorescence on the sea, and the spinning planets and the expanding universe, all are bound together by the elastic string of time. It is advisable to look from the tide pool to the stars and back to the tide pool again. It's probably one of the most quoted really in Steinbeck's book. Generations of marine biologists have taken this into heart. So looking at the fish, try to get to know them. Sometimes it's really fun to go snorkeling. See this convict surgeonfish feeding on the algae. I can put a Latin name on that, Acanthurus triostigos. And being a fisheries major, I should be able to count its spines, but there's more to counting its spines. This tradition of self-reflection when we're doing our work as marine scientists even continues to the present day. An oceanographer and a captain, Diana Zuniga from Galicia in Spain, writes, if you are a marine scientist, it could be thought that isolation is not the perfect environment for conducting marine research, when it is expected to participate in campaigns on board, research vessels, or work in a crowded laboratory. Well, nothing is further from the truth. During solitude, you can read and, most important, reflect on what you have read. In my humble opinion, I always thought that this is the best way to do science. You have to go and reflect. And the way to reflect on reality and the whole of nature, one of the best tools that we have is literature. And so there's still this tension about the arts and sciences. Okay? Isaac Newton, a philosopher, on a critique of critique and his take on the Log of the Sea of Cortez of Steinbeck and Ricketts. He says that the Sea of Cortez is this clash of worldviews between the scientist content to focus his energy in the tide pool and the stars, and the novelist who seeks to know human hearts and minds, and that innate desire that we all have to reach out and connect with others. Ricketts' philosophy is profound but detached, fit for a sage who shields himself from human concerns in order to seek enlightenment. Whereas Steinbeck's intense compassion for the plight of humankind can come across as overtly sentimental. I know on a personal level the appeal and perils of both. The interesting thing here, and as I end this talk, is that Ricketts was trained in the sciences, but loved philosophy and the arts. While Steinbeck, the writer, was already a well-known novelist when he went back to marine biology. And yet wanted to know the scientific. So wanted to get a better grasp of the scientific. And so they came out with the work, which is quite remarkable in the history of marine science. A Nobel Prize winning writer and a marine zoologist coming up with one of the most beautiful prose ever written in the genre of the essay. So I think with that, I could end this talk. No, hopefully later on there will be questions, and we'll be willing to answer whatever is what you want to ask, and probably if you have an opinion, we can have a discussion later on. So again, thank you and good afternoon to all.
Thank you so much po, Dr. Ben. And our next speaker is a professor at the Institute of Biology of UP Diliman. She obtained her PhD from the Department of Immunology, Microbiology, and Parasitology of the Louisiana State University Health Science Center in New Orleans, USA. During her doctorate, she indulged in the rich culture of arts, music, and poetry in New Orleans as a spectator while persevering in her training as a scientist. When her time permits, she paints symbolic flower art, informative children's art, and doodles flower shapes for a relaxing weekend activity. She habitually takes photos of flowers and has been fascinated by the biological processes that occur in them. Here is the Institute of Biology's Dr. Joyce Ibana.
Salamat. Maraming salamat for your very kind and generous introduction. Good afternoon, everyone, and thank you for the College of Science Communication team, AD Lilian and the team, and also the administration of College of Science for giving us the opportunity to share with you our journey in science and arts. So as previously mentioned, what I have in store for you is a discussion on a journey in science, arts, and innovation. And I will also share my hypothesis on how these three could come together in synergy to make more social impact for our work. So I take you to this journey and I share with you a personal journey so that when you ask questions about the thought process, I can actually give you an insight of exactly what happened in my journey as a scientist, arts, and an innovation officer for some time. And this journey is started with science. And what I can share with you on my thoughts about these three is that while science is more straightforward, I think the journey is you work hard, you publish papers, and you get rewards. It's relatively straightforward. Unlike art, which I find to be more chaotic and uncertain, and there's a lot of unknowns and human factor. But this is when I was more in touch with my humanity. This is when I am more in touch with the pains of and joys of lives. And in innovation, what I noticed is this is a continuous iteration of prototyping, coming up with new, better versions of previous products or thoughts or ideas, and a continuous process of making things better. So let's start with science. My journey in science started in here. This is a school in Daet, Camarines Norte, Mabini Colleges. And when I was a young girl, I used to tag along my parents, who are both educators. My mother is a grammar instructor and an English literature teacher. And my father teaches mathematics, science, military science, and agriculture. But during those years, in my younger years, I tag along with them. I always speak at this chemistry laboratory in this school. And I was able to visit it recently, and I took this photo. And what I saw there is that the different cool looking glasswares and the fascinating changes in colors when there are changes in chemical reactions. So it was something that I was very fascinated, that when I graduated in preparatory school, my parents wanted me to become a lawyer, but and usually in the province, most kids, parents would want their children to be doctors, lawyers, and engineers, and what have you. But when I stood there, I blurted out I wanted to be a chemist, to everyone's surprise. And this was inspired by what I saw of the beauty of the experiments that were done in the chemistry laboratory. And many years later, I pursued and I was hyper focused on becoming a scientist. And I ended up to become a cell biologist and an immunologist. And I spent a lot of time looking at a bacterium that lives inside a cell under the microscope. So this is a confocal microscope, a very high-powered microscope that can actually delve into the different layers of a cell. So this is what happened. So as a scientist, I just realized recently that my motivation was actually the beauty and the colors that come along with doing experiments in chemistry. And this motivation I carried on until I pursued my PhD. But I was always fascinated by how things work, and I'm always fascinated with how nature works. And I study and focus on cell signaling pathway. So cell signaling is when you get a signal from the external environment, the cell would have a cascade of biological reaction that would result in the response that would permit the different biological function that is required by the cell. But this can be viewed under a confocal microscopy, which is beautifully seen when you use fluorescent microscopy. So this is actually a nucleus of the cell, this blue. This is the bacterium that I study, Chlamydia trachomatis. And this is a protein that I am interested in, the STAT1 protein, and this is colored red. So it's fascinating how to understand the pathogenesis of this bacterium, how it manipulates host cell biology, and you can visualize this under a powerful microscope. So for some scientists, they focus on studying birds, or in other cases, as mentioned earlier, starfish or other marine animals, or flowers and other natural biological species. But I just happened to be fascinated by a bacterium called Chlamydia trachomatis that it resides inside a human host cell or other cells of animals. And we do a lot of sophisticated analysis. We could study this cell and in confocal microscopy and study a single cell, and we look at the expression of proteins, where they are located, whether they are in the nucleus or in the cytoplasm. So it's really, really fascinating. And this is from one of the papers. And what I actually realized is that for most of my papers on chlamydia, I always end up with a conceptual map. So whenever I study a certain mechanism and investigate how things work, I end up with a model of illustrating how things work. And this is from one of the papers that we published. And this always happens to me. Even this is another paper published in Infection and Immunity. I always illustrate what is the overall story of the data that I have generated and the interpretation of the data that I have written. I always do this. And I carried on this practice in mentoring my students as well. Whenever I ask them, so what's your story? What happened to your data? What's your proposal? I always ask them to make a graphical abstract, because I believe that we understand things better when we can illustrate things. And one of the PhD work that I completed in Chlamydia trachomatis ended up as a cover of American Society journal Infection and Immunity. So it was in 2011. I was very, very proud of this work, and I was so happy because I dreamt to become a scientist since I was a kid. And after being in the US for 8 years, my mother visited me, and I proudly showed him my accomplishment. And to my surprise, my mother asked me, so what did you get out of this? And it was the most difficult question I've been asked in my whole journey as a scientist. What did I get out of this? And it's a question that I didn't answer at the time, but I know that my mother is a very wise and a mother who knows what is best for me. And that question prompted me to accept the invitation of the University of the Philippines to come home in 2013. I started searching for what did I get out of this. I pursued science. It was my childhood dream, but my mother asked me, what did I get out of this? And coming home in 2013 was one of the wisest decisions that I have done in my life, I think. So it's 10 years now. And it's a story of my journey from being focused from molecules to man. Because here I found the joy of service in helping other younger generation of scientists to realize their dream to become a scientist. At least for now, I think I realize if that would answer my mother's question, what did I get out of this? I find a joy in teaching, just like my parents did. And now we go to the journey in the art. In 2017, I encountered, as many would have perhaps, a midlife transition when you start questioning. It started from the question of my mother, and I started questioning other things that I'm doing in life. And I wrote this poem, which is titled "A Farewell to the Simple Beauty of the Flower." And I will read this to you. I have always admired the exquisite beauty of flowers. When I was a child, I stared at them, smelled them roses, daisies, sampaguita, rosals. I even waited for them to bloom in the middle of the night and watched them fade away before dawn. But 40-something years later, they have ceased to be simply flowers. They have become overwhelming sources of wonder. I can no longer sit in silence admiring their beauty without questioning why, how, what made them bloom, and what happens after. The curse of a scientist. Although in my eyes, flowers have maintained their beauty, they have lost their simplicity. And this is a story that resonates with other scientists. We come to a point sometimes when we investigate a particular organism of interest and we become overwhelmed by the many things we know about them, and then overwhelmed as well by the many things that we still don't know about them, and to the point that we question, so what is this for? And so this journey continued, and I became more in touch with the pains and the joys of doing science, as well as the frustrations. And also I become more in touch with myself and my real love for arts. But when I went back to what I studied in many years as a researcher, as a cell biologist and an immunology, something occurred to me later on in life that I felt a lot of things were irrelevant unless we are able to communicate them to the public on how it can impact human lives. Because this organism that I was studying for many years, out of the fascination of its beauty and the mechanism of its pathogenesis, is actually a very important pathogen in the public health perspective. It causes a silent disease. It's a sexually transmitted infection that can cause infectious blindness if it infects the conjunctiva, and tubal factor infertility if it infects the urogenital tract. It's the world's most common sexually transmitted infection because it's mostly asymptomatic and therefore undetected and untreated. But many do not know that this may increase their risk for HIV/AIDS and cervical cancer. And sadly, this can also be vertically transmitted from mother to infant during vaginal delivery and can cause the innocent children infant pneumonia and neonatal conjunctivitis. So this realization that I have been studying chlamydia as a basic scientist and not able to communicate how this impacts human health and families and women's and children led me to really felt a strong desire to communicate better the impact of science to humanity. But I didn't know how at the time because I don't have the training, but I continued to paint flowers. And interestingly, these paintings that I started to do, there was a entrepreneur. The name of is Nympha Salazar, which I accidentally met in the common room here in Katipunan. And she was interested in the flower paintings that I made, and she said, "Why don't we put it in my product?" It was an infinity shawl. So it was just out of the blue. I did not expect this to happen. It happened in 2019. And we collaborated and she put this in this infinity. This is just one infinity shawl that can be worn in different geometric shapes in different styles. And it looks so, it really was fascinating how my collaboration with local industry evolved into this. And I was surprised that when this happened in September of 2019. And during the Christmas season, she was sending me pictures of many people buying this as Christmas gift. And I was so surprised, and I did not expect this to happen. And in February of 2020, so we have some. I wasn't planning on having this venture in painting as an entrepreneur. I have no concept of entrepreneur, and but during that time, she said, you have accumulated this much for this intellectual property of your art, but pandemic came, and in 2020. It was fascinating that I have this without costing me anything, the ability to donate this work so that the workers who are involved, because all the stores were closed and so the sales went down. So there was this ability to have something to give to the local industry, and this is fascinating because in science, our impact is based on the citations. So when other people cites the published work and turn it into another publication, and one of my most cited paper have 178 citation that happened over 2060, about 7 years. But this one happened, you gave joy to many people, 500 people in just 3 months. So it's very fascinating how art can actually impact people in their small, in a very in my, I consider my small way, but it really makes an impact in the economy. And also I was inspired by that experience, and I continue doing arts, and I also do children's arts during the pandemic, so that to communicate the importance of or holistic health. I think the value of good night's sleep is underappreciated. The value of exercise is underappreciated in our need for a strong immune response against many infectious diseases. And I came up with this children's arts to communicate this. And also I did this COVID arts and immunology. This is my depiction of how the RNA virus have actually caused a lot of suffering in the world, and the coronavirus. And this is actually based on the structure, pentamer structure of the IgM antibody, and this is the coronavirus, depicting the significance of our immune response. And this is the structure of IgG. So compared to the pentameric IgM, here is the structure of IgG. And this is actually a representation of the adaptive immune response. This represents the dendritic cells and the arrival of the T cell response. So this is how I actually process my scientific mind when I'm trying to relax. So I don't believe that there is actually a left brain or a right brain person. I mean, we all have both brains, and we actually use them, maybe not in painting, but creativity and technical skills are both important for everyone's work. I think we have to shift from a wandering mode to be creative, and then to a executive mode to do the specific structured function. So I don't consider those as two separate realms. They don't perhaps function simultaneously, but you can switch back and forth in these two modes. And I continue this, and I take inspiration from when I have some thoughts or some sadness or some when I lose hope. I contemplate on the beauty of the flower, and this is one is some a flower that you will find at the pond at the back of the Quezon Hall. You will see this, and one after the pandemic, I was walking there, and I guess most of us have experienced this lack of hope and really depressing times in that time. And but when I look at this flower, I realized that it was fascinating that even if it's surrounded by muddy pool or this water, it dutifully rises every day to rise and face the sun, the wind, and the rain. So I think if the flower can do this, why can't we? Why can't I? So I entitled the painting Ang Muling Pagbangon or Pag-ahon. And it helped me process all the difficulties that we went through in the pandemic, and that how with the inspiration of the flower, we can always face whatever is there to face, to do our duties in service of our country and our fellow men. And this is another a painting where I process the pains of doing service for our country. It's really difficult to be a scientist in our country, and but this painting is based on the idea of the oysters making pearl. You know how it starts with the invasion of a grain, which is a painful process for this organism, but it results in a very valuable and beautiful creation like the pearl. And I think that our love for our country symbolizes like that. We can use it to create something beautiful and valuable for everyone. So I embrace that in this journey from science to art. I embrace that deep inside me, I'm also an artist, and I started this journey with the help of Mr. Fernando Sena, who's actually helped me in painting using acrylic. And this is from last month's group exhibits in his gallery. So I thank him a lot for recognizing that I can do this as well. So finally, in the innovation part, I was fortunate and I was given the opportunity of the College of Science to serve in the innovation office for some time, and also the UP system as a university innovation fellow. And I learned a lot about how to serve in the innovation scheme in terms of providing innovative educational tools. These are arts that are done by our students. They're representing the IgM molecule using materials that are, this project is like sining sa basura. So we make this creation out of recycled materials from the lab. And our arts also has been used as for the scholarship program initiated by AD Lilian at the Institute of Biology Alumni Association. And finally, I will venturing into real data arts, where we are going to use real data as an art for our advocacy in communicating the importance of understanding the infection, chlamydia trachomatis, and hope that it will actually prevent this infection and the many diseases that it causes. But I am still in the process of refining this venture, and one of the questions that I need to address is how do we actually increase the social impact of an innovation? So I will end this talk with my hypothesis on how this intersection actually work together. So most of us here are very familiar with science. I said it's simple. You do your experiment, generate data, relatively simple, generate data, you publish paper, and then you contribute to the generation of knowledge. But then we are called to solve problems, real world problems, and therefore we are called to focus as well as some efforts on innovation to address real world problems. And there is where we actually have the intersection, where we have the invention. So why is it important that we also cultivate liberal arts? I think with science and innovation, we can have a lot of good technical solutions. But if I ask you whether technical solution is sufficient to solve problems, you can look at your bag right now. And if I ask you what's the function of your bag? And perhaps you will say, well, it's for carrying my cell phone, my wallet, or my notebook, or what have you. But that's a function. But a plastic bag can do that. If you look at your bag, you will realize that your decision to buy that particular bag was based a lot more on your humanity, on your favorite color, of the local, perhaps local community that you want to support, a brand, or something else that relates to your humanity. And therefore, I think that there is important to integrate arts in science and innovation. Because there are many experts in science, but you will also notice that those who are most or making more impact are those who can artfully communicate their science. And what I learned also recently, as in my training as an innovation university innovation fellow, is that the integration of arts and innovation is also important in marketing your ideas. Because whether you have a very good technical solution, if you cannot change the behavior of people to actually adapt your ideas or your invention or your product, it will be difficult to make social impact. And so putting this realm of art, science, innovation in a nurturing and enabling environment, I hypothesize is what would make us, allow us to make better social impact. So with that, I'd like to end my talk, and I'd like to thank the organizers of this event, and I also would like to end with the advocacy for embracing diversity. This is a painting that I made, the third painting I made from with Mr. Sena, and I remember one of my favorite saints, Saint Therese, mentioned something like, "Not everyone have to be roses. We have to appreciate the daisies, the lilies, and everything, because that's how the world is beautiful." And I am very fortunate as well with the College of Science, UP Diliman, and UP System, and the Institute of Biology for embracing our diversity and allowing us to become scientists, innovators, and to pursue whatever we can contribute value to our university and to our country. And to everyone who is here, I'll share with you this prayer for daughters. And I hope that everyone, I just mentioned, this titled daughter because I wrote it, but it could be anyone. And I pray that just like a rose with thorns, may we stay as gentle, kind, and sweet as we are, but may we have the courage to not let anyone deprive us of our freedom, to fight for our happiness, grow to our full potential, keep our self respect, and be who we are meant to be. So, for those who want to be an artist, a scientist, and an innovator, I wish you all the best. I think you can do it. Thank you.
Maraming salamat po, Doc Joyce. Thank you. That poem was very lovely, and thank you for blessing us all with it. I'd like to say, ma'am, sobrang natuwa po yung audience po natin. There's someone in the comments who actually said they love your paintings, and I think this would be a great time to just make a quick segue po sa mga nanonood. There is still an ongoing exhibit at the Institute of Biology. Featured po yung mga paintings ni Ma'am Joyce, ni Doc Joyce, and a lot of other people from the Institute. Hanggang sa katapusan na lang ng buwan po siya. So you still have a few days. Please come anytime. It's free, free to the public. Para makita niyo rin po yung napakarami pong talents at yung napakagandang artwork na nagagawa po ng ating mga scientists. So, at saka before we continue, I'd also like to remind our audience na meron po tayong Q&A at the end of the entire session. So please feel free to drop your questions into the Q&A box. Sa live stream, those who are following us on Facebook, please, you can comment there. We will look at those later on. So without further ado, let's proceed to our next speaker. Siya po ay Associate Professor of Chemistry, and her research is focused on LC and MS-based metabolomics. She's also the manager of the LC-MS facility at the Institute of Chemistry of the University of the Philippines, Diliman. She has a very active team of researchers and graduate students working on LC and MS-based metabolomics of terrestrial and marine organisms and microorganisms. She aims to create a laboratory of small molecules from a library, to create a library of small molecules from Philippine flora and fauna metabolomics. Ladies and gentlemen and everyone, please welcome Dr. Hiyas Junio from the Institute of Chemistry. Ma'am, nakamute po kayo?
Yeah, hello, good afternoon. Let me just set up my presentation. Okay, so good afternoon, everybody. It's a pleasure to be here and to talk about my work as a scientist, and as well as how art actually inspires us. So it's actually, the title is How Scientists Use Art to Explore the World, but it's actually how art inspires us to do our science. Just to be honest with everybody, confession time, I guess. Being a scientist was never my first choice for a career. It's actually being a painter was my first choice, but I was sort of discouraged to take up fine arts because apparently it's not very lucrative. So to my dismay, I said, okay, I'll settle for science. I don't know up to now if whether that was a wise decision. But in any case, I would like to talk about our work. The introduction actually didn't say a lot how we use chemistry in order to study about art or to express art. So I'm going to talk about chemistry and color. When I was in grad school, I always have this moment where I feel like it's getting overwhelming to do the work. So what I would always do is I would take my camera and then just walk around campus to the point that I already memorized the plants that are growing inside the campus. And I know their time when the flowers would bloom or where the birds are supposed to be. So one of the outlet that I did just to manage my stress in grad school was to take photos. So these are some of the photos I have taken in grad school. I just take photos and then I go back to it and then take some more and then go back to it until I amassed a significant number of photos. I kind of stopped taking photos since I got back from the Philippines to be a faculty, but then I came across all of my old photos last night when I was looking for something to put in the presentation. And I said, well, this is all about colors, so I would like to share my point of view then when I was taking the photos, and I was really, to a certain extent, attracted to the colors in my surroundings. So I feel like these are the ones that really influenced me in terms of the research that I currently am doing. So the research work was partly inspired by my graduate students, and I think we kind of stepped into the rainbow in this sense, because we wanted to study natural dyes. So one of my previous graduate student, Mr. Molino, he said, I would like to study on the natural dyes and the tradition that goes with studying natural dyes. And he used this book, Gampol, which was actually written by my classmate in undergrad in UP, and it's all about it's a compendium of Philippine natural dye yielding plants. So we use this as a reference for his master's thesis, and as you can see, there's a big surge in the demand for natural dyes and textiles. So it comes it not naturally comes with that the demand for the use of natural dyes sourced from our natural resources, plants, even microorganisms as well. So that's where we kind of started getting into more on the chemistry of natural dyes with my student, Mr. Molino. So I will talk about most of his work, and then I'll also talk about some of the work, future work. So this is actually a picture of some cut out jeans or samples of denim that were dyed using the Japanese indigo. The Japanese indigo is derived from a plant called Persicaria tinctoria. And as you can see, they have different shades of blue. And the different shades of blue comes from the different processes, which is basically, the word just escapes me, fermentation. Different fermentation processes will give different colors of blue. In the Philippines, we also have a local community who produce indigo dye, but it's from a different plant. It's called Indigofera tinctoria, and this photo of this plant was taken from Abra. Okay, so one of the things that we wanted to do in collaboration with Philippine Textile Research Institute is to actually assess the quality of the indigo natural dyes that are produced by the small community of people who produce this dye. So as you can see from this picture here, they are all considered or sold as indigo dye, but as you can see, they have different colors. Some of them are more pinkish, more purple, and some of them are quite yellow. So it's not really indigo dye, but it has something to do with the chemistry that's happening when they produce the dye. So it's a common misconception that when we say indigo, we think it's actually a single color, but it's not. It's actually a mixture of different colors. And this mechanism here actually shows you the process that the molecules making up the indigo dye, the process they undergo, why some of them are blue, some of them are purple, and some of them are quite yellow. So through the process of hydroxylation, oxidation, and some degradation, you actually get to have indigotine. Indigotine is the blue component of indigo. And then we also have indirubin. Indirubin is more on the reddish purplish color, and then we have also isoindigo. This is more on the orange and yellow part of the color wheel. So if you imagine you have the mixture of all of these three in varying amounts, the sum total color of your indigo dye would definitely be different. And we wanted to make sure that the ones that are being produced by the local community is that it has a higher content of indigo rather than indirubin, so that the color will be bluer as opposed to more purple. So we wanted to answer that question using our technique that we use in the lab. So this is now more on the chemistry part of the indigo production. So as mentioned, my research work focuses more on liquid chromatography, mass spectrometry, and we try to analyze small molecules. So most of the work that we do is we just try to analyze small molecules from different sources, be it marine sample, plant sample. We actually even analyze honey samples and so on and so forth. So basically, we take the plant or the microorganism and we extract it. The extractions we use for natural dyes follows the extraction procedure suggested in by the PTIR handbook, and mostly we also extract it using water because water is a good solvent, and we know that it's not harmful because it's basically water. So we want to start from something that is not harmful to the environment. And then we do what we call a full scan analysis and what we call a tandem mass spec analysis. So I'm not going to go through that anymore, so just so you have an idea, but I just wanted to show you, this is the instrument that we use. We have this at the Institute of Chemistry. So when the samples go through these instruments, they get detected by this machine here. This is called the quadruple time of flight, and it actually measures the molecular weight of a particular molecule. And then as a simulation, this is what you actually see when you detect the molecular weight of a particular molecule. To be exact, we're detecting ions, but from the ions, we can identify the molecular weight of a molecule. So from this data, right, basically what happens is that the molecule is broken down into pieces. Okay, and from those pieces, we are able to identify the particular structure of the molecule by using a reference library. So here, so you can see here from the top, this is the broken down pieces of the molecule. And from at the bottom, you can see the reference molecule also broken down into pieces. And since they match, we are able to identify that the particular molecule that we have detected corresponds to this particular structure, and that belongs to a group of compound called flavonoids. So that's the science that we do. It also involves art in the sense of how are you going to put back the pieces together to identify the structure of the molecule. Okay, so we were able to publish this result in the Rapid Communications in Mass Spectrometry. I was so proud of my student because he was able to publish this before he graduated in his MS degree. So this is the result of that analysis. As you can see, you have two compounds, compounds 1 and compounds 2. For the chemists out there, if you look at the label of compounds 1 and compounds 2, they have exactly the same molecular weight. And in chemistry, we call these two compounds as isomers. They are isomeric, meaning they have the same molecular formula. They have a different structural formula. Okay, in this case, it's actually just this part here that are different between these two compounds because they are flipped. That's the only difference. But by using our method, we were able to distinguish the indigotine and indirubin. So by showing this, we would be able now to assess the different types of natural dyes, the indigo powder that are being produced by the local communities. So with that, we are now in part of our work actually is to identify how come, right? These are samples given to us by PTRI of cloth that were dyed using indigo from different suppliers. So they wanted us to know why one is more blue than the other and one is lighter than the other. So this is currently being done. So hopefully we get to finish this research and then we'll get to publish, and then this will help to standardize the indigo products that are being sold here in the Philippines so that we can have a competitive edge and we can sell those indigo dye powders outside of our country as a means of economic gain. Okay. And so we kind of wanted to expand that thinking that we can have more sources of natural dyes, not just indigo. So we went on and looked for some sources of natural dyes. We call this the plant Pantone because we were able to produce some colors that are really vibrant. And we looked at some of the endemic plants that we have, in particular, Dillenia philippinensis, Diospyros blancoi, and the Eucalyptus deglupta. We also wanted to study Syzygium species such as these two because they're not very popular as like a dessert fruit. So we thought maybe we can find something that we can do with the Syzygium species, because when it's fruiting season, there's a lot of these fruits that sometimes they just fall onto the ground. So maybe we can find something to increase the value chain of these particular fruits. And then also we used Terminalia catapa, which is talisay, and talisay is a known source of natural dye. So we use it as some sort of reference for our study. And as you can see, when we extracted the natural dyes using water, so this is just simply boiling this samples in water, we were able to get a whole range of colors. They're more on the pastel type as pastel color palette. Okay, so these are the colors that we were able to get from this plant samples. Right. So okay. So basically, we wanted to see if these dyes will also work when we combine them with or when we use them as text for textiles. So we had to make an experiment, and then so what we did is we mixed them with different mordants. So mordants are actually used in the textile industry so that the dye would stick more to the fabric. Okay, and in terms of the chemistry here, it's mostly coordination chemistry, meaning that the metals in the mordants will interact with the molecules in the dye, and then that's going to create some sort of reaction, resulting into changes in color. So as you can see here, this is, so just to go through the slide, the first column is actually the result when we mixed the decoction with ferrous sulfate. The next one is with alum, the next one is with copper sulfate, then tartaric acid, and then soda ash. And then as you can see, by changing the type of mordants that we add, there's a subsequent changes in color, which means that there's a chemical reaction that's actually happening in the solution. Okay. However, when we try to use this with textiles, in particular, these are cotton fabrics, you can see that not all of the colors in the solution actually translated well into the fabric. And again, the reason for that is because of chemistry. It means that some of the molecules didn't really have a strong interaction with the fabric, such that when you wash the fabric, the colors are just washed off with the water. So I think that's what you call color fastness. So it means that the colors do not stick to the fabric, right? And it has something to do with the solubility of the small molecules that are in the fabric. So it's either you explore other sources of natural dyes or you use different types of mordant that will allow the small molecules to stick better to the fabric. But one of the things that we were fascinated about is the production of the black color in the fabric from Dillenia philippinensis and Idigulupta bark. So because Dillenia philippinensis is not really something that is used as a source of natural dye. If you're not familiar, Dillenia philippinensis is also called katmon, and katmon is used mostly as a souring agent for sinigang or some sort of like to make a soup sour, right? So it's not actually very popular to some of the people in Manila. Actually, if I ask people around, do you know what katmon is? They don't know what katmon is, right? But we have very beautiful katmon trees that are on campus as well as along, I think, the BIR road. There's a lot of katmon trees. Also in UP Town Center, there's a lot of katmon trees there. But in any case, we wanted to look at the difference in terms of sorry about the noise. the different color, the intensity of the black colors produced by the Lena philippinensis and the Idiglutta bark. And we saw that tannins are the ones that are responsible for the production of the black color. And based on our analysis, right, some tannins are exclusively found in Idiglutta and some tannins are exclusively found in Katmon, but there are some tannins that are found in both plants. So we think that this particular compound here, procyanidin B2, is the one that is responsible for the darker color of Katmon present in Idiglutta. Similarly, there's also some black color produced by the talisay leaf and also by the Diospyros blancoi. This should be spice blanco. I forgot to change it, or mabolo fruit. And then we saw that in terms of the size of the tannins in the talisay is larger compared to the tannins in mabolo, which is smaller. And we think in the size of the tannin also has some sort of effect on the type of black color that is produced in the fabric. If you notice the one here for the mabolo, it has kind of like a bluish tinge to it, but we haven't really looked into that as of yet. We also went and looked at the yellow yellow pigment produced by both the talisay and the katmon when they are used for the textiles, and we found out that they have compounds that are common to both of them. The colors are almost similar, so we would think that some of these molecules here that are common to both talisay and the Lena philippinensis are the ones that will be responsible for the production of the yellow color. All right, so having said that, we would like to produce more natural dyes, but then again, you have to harvest the fruit, harvest some of the bark, and harvest the leaves. Although some of them are abundant in nature, we were thinking of something more sustainable in the sense that it will be easier to collect, easier to produce in a large scale. That's why we wanted to explore plant endophytes. So what you're seeing right now are actually plates of fungi. So these are fungi, and these are not the typical fungi that you see that grows out of your bread, moldy bread, right? So they have different colors. And these fungi were isolated. I was able to isolate these fungi from different plants. And as you can see, they sort of have like some growth rings, and that is basically functions like the growth ring of the trees. It actually tells us how old this plate culture is. I think this is about like 3 days per ring, but it varies. The what do you call this? The age of the ring varies per particular fungus. So these have already been. These are isolates, so these are pure isolates of this fungi, and as you can see, they have different color. This one has orange, green, and orange, purple, and a little bit of pink. This one is more light yellow and brown. This one also has a tinge of pink and a little bit darker color. So the first photo I showed, those are grown in media plate, but sometimes we try and grow them in liquid culture. And if you grow fungi in liquid culture, you would notice they would have this circular sphere. They look like small milk bubble tea, right? When you grow them in liquid culture with continuous shaking, so they look like bubble tea, and they also have different colors. So fungi, they grow inside the plant, and they have a symbiotic bonding to pathogenic relationship with the plants, which means these fungi do not actually cause plants to have disease. So they have like a symbiotic relationship, but some of them almost bordering pathogenic, but not enough to give the plant any disease. So they are living, thriving inside the plant. So these are some of the dye or natural pigments that we were able to isolate from this endophytes. And as you can see, they're very colorful, they're very vibrant. And they vary in terms of they have green, we have greens, we have browns, yellow, pink, so on and so forth. So which leads me to my very first obsession. It's kind of, I think it's an obsession with this particular endemic plant, which is Eucalyptus deglupta. The reason for this is because I was really fascinated when I saw it, because I've never seen a tree with a trunk as colorful as Eucalyptus deglupta. So this is called rainbow tree, and this is called Mindanao gum tree, or local term bagras. Although we discovered that bagras can refer to another type of tree. So bagras, maybe it's used in different region, but it doesn't necessarily refer to the rainbow tree. In any case, the first one I've seen it in Mindanao, and it was larger than this. When the tree is larger, it produces a whole lot of different colors, not just orange, but you will see blue, tinge of blue, and tinge of purple on the bark. Okay, but my curiosity was actually piqued because of the color of the bark. So my question here is, what is the science behind the rainbow color of the bark? And are endophytes involved in the production of this pigment? So basically what we did is we tried to investigate that, and we actually isolated some endophytes from the rainbow eucalyptus. So these are some of the endophytes that we were able to isolate from the tree. There is a particular endophyte that we're really interested in, because it's the one that produces the same orange color as the one produced by the tree. However, because of the pandemic, we were not able to care for the endophyte, and it died. Okay, so we were not able to isolate it and grow it in larger quantities to be able for us to identify. And it also grows only at a specific time, or we can isolate it only at a specific time of the year. We can only grow it in March. So it will be March next month. So we will try and see if we will be able to find it again. But our idea of studying, looking for endophytes, looking for microorganisms to produce natural dyes is more on sustainability, and as well as it's easier to culture fungi for large or for upscale use. So our idea here is to collect a lot of these natural dyes from microorganism, and we are talking with, or we had discussion with UP College of Fine Arts that when we collected a lot or enough sample of our natural dyes, that we will give it to them so that they can use it for their artwork, for painting or whatever artwork they can create out of those dyes. So those are the goals of this particular project. And with that, I would like to thank our audience for listening, and also, of course, to thank the funding agencies that actually help us to do our work, which is basically chemistry and colors. So thank you, everybody.
Thank you. Thank you so much, Dr. Yas. And our last speaker is a botanist specializing in plant development. She teaches plant morphoanatomy and plant developmental biology courses at the Institute of Biology. She is also currently continuing her research on the biology of Rhodesiae. She only started painting during the pandemic and considers painting as a very effective therapy for her anxiety. She uses watercolor and she paints mostly flowers. She equally loves doing loose florals and very detailed botanical paintings. She takes her time painting the smallest details for botanical artwork to ensure that the floral structures and morphology are accurate. And just in 2021, she joined an online botanical art exhibit at the Philippine Botanical Art Society. Let's hear from Dr. Erika Bascos.
Hello, good afternoon, everyone. All right, so is my audio okay? Yes.
Okay naman
Okay.
po.
All right. So again, good afternoon. so excited to share with you my journey towards becoming a so-called scientist artist. So how I went from just studying the flowers to teaching about them to now painting them. And I would also like to touch on how art actually helped me deal with anxiety and depression during the pandemic. Right, so this is me studying this particular plant, which I'm sure many of you know. So this is Rafflesia. It is known for its smell, which is like death. All right. So it just so happens that this particular plant species only produces flowers. So it doesn't form any root, stem, or leaves. So just the flowers. So this is the plant that I'm studying. So how did I go from studying flowers to painting them? Well, it's actually quite simple. All it took was one global pandemic. So I actually consider myself as an accidental artist because I never would have attempted painting if the pandemic didn't happen. So prior to 2020, I never really did anything painting related. So I was never an artsy person. And the only time I ever drew or sketched something was during the time when I was a student, when I had to draw plant specimens in our laboratory classes there. But even though I never noticed it, I'm always surrounded by botanical drawings, botanical illustrations. Like, for example, in one of the papers that we published on Rafflesia, we had to hire an illustrator to make this beautiful drawing of the Rafflesia flower so that we can show our readers its morphology, the different structures, because this is very important in the taxonomy of this genus. And also, in the different subjects that I teach. So I'm a teacher at the same time. I was also a student in all of these different courses. So during the time when I was a BS Bio undergraduate, I also needed to draw all of these different specimens that we show during our lab class. So what we do is we lay out all of these different plant specimens and we ask our students to draw them. And of course, during our time when our cell phones didn't have cameras yet, and only one or two of our classmates had digital cameras, we really had to rely on our drawings to remember the different structures and the plants which they are associated with there. So, and up until now, we still ask our students to do this. But of course, now they have cell phones with cameras so they can take pictures, but we still ask them to draw because we feel that if they take the time to really study the specimen and look at the different structures, that's when they remember all of the different parts. And some of my classmates, I remember, and even some of my students later on, they would say that, oh my God, we thought that this was Bio 101. It turns out it's Fine Arts 101 because of all of these different plant specimens that they needed to draw there. And we also noticed that the students who really took the time and made the effort to draw the different specimens properly, they're also the ones who got really high scores during the MOVE type laboratory exams there. So, but of course, we do not grade them based on how well they draw. We actually grade their plates based on how complete their drawings are, if they were able to identify and label the structures properly. So if you look at this is, singkamas and carrot, but art there. So imagine what the students can do using just a pencil and paper. So these are the actual drawings of some of my former students. They're now already doctors, So I really had to contact them to ask for permission if I could post their drawings. So here, so these students, even though their drawings are really very good, they didn't get perfect scores because they missed, some of the labels there. So it's really very nice to look at how the students see this specimen. So this is how the students see kamoteng kahoy with their own eyes. And their interpretation there and the picture plant. It's really very nice to look at these. That's why I kept these plates for a very long time, parang more than a decade already. And sometimes when we cannot bring the specimens to the laboratory, we ask our students to do a tour around campus in order for them to see the different plants, and they're still required to draw them. And this helps them remember really the plants, especially in taxonomy. There, if they drew the different parts of the plants, then they're sure to remember it during the exam. And not just the macro specimens, even the ones that they see under the microscope. Some students, even though we ask them to just do diagrammatic drawings, some of them really love drawing everything cell by cell, like this one. Maybe it helps them remember all of the different parts there, but we do not require them to make drawings as detailed as this one, because imagine the amount of time and effort this takes. And this is just one specimen. And in one laboratory class, they're required to draw 15 or more of these in the 3-hour laboratory period there. So all of these things, all of the botanical illustrations, of the drawings that I see in my research, in my teaching, I believe that this really helped me, in my sketches when I do my work as a watercolorist. So I am super concerned with the details. If I am going to draw leaves, if that particular plant has stipules, then my drawing has to have stipules there. Like that. So I am super bothered by the phyllotaxy, if the arrangement of the leaves in my drawings are accurate or not. So that's how it is for me, because of all of the different drawings that I saw from the time I was a student to the time that I am now teaching all of these plant laboratory courses. And before I go to that in more detail, let me first tell you the story of how I got to start painting. So I remember the exact date when I started painting. Prior to this, I have never touched a paintbrush in my life. So in June of 2020, it was 3 months into the pandemic, and my 2 daughters, of course, were getting extremely bored with, staying at home for more than 3 months. And they were sick of their toys, they were sick of Netflix, and they were looking for something new to do. And they asked me if they could paint. And of course, the easiest, art material to get is watercolor, like something. I think I bought something like this. I bought it for 70 pesos. It was with a brush already there. And they started painting, and they asked me to join them as they paint. And my youngest, who was turning 4 back then, she asked me to paint some flowers, which was a good thing, because if she had asked me to paint something else, like maybe a Roblox character or some apes or giraffes or whatever, I would have been in real trouble. But since I know flowers, I was able to make these 2 paintings. So these were my very, very first paintings. And I remember I posted them on my Facebook page, and many of my friends commented about, how good they were. I didn't know you could paint. These are amazing. You could sell them, all of those things. And of course, in the next few days, I found that I really liked doing it. So I painted more. These are just from my imagination. So I didn't use any reference photos. So these are not real flowers. So please do not ask me what species these are. These are from my imagination there. And I started painting more and more, maybe one for every day. Some of them I posted in Facebook. Some I just kept for myself because I didn't want anybody to see. So I painted this one for my daughter's 4th birthday, and this one for another relative's birthday. So there. And then, people would always would have really nice things to say about my paintings. But of course, that's not the reason why I paint. So I really continued painting because it really helped me deal with, the anxiety brought by the pandemic. And I had a lot during that time. And I remember that it was the first time that I felt that I needed, professional help to deal with it. And I remember the psychologist from PsychServe who I consulted, she said, you need to break the routine. You must do something new, something different to make you feel fortunate that this day happened. And this was it for me. It was painting for me. And I remember I posted this back in July of 2020. And I said that painting gave me, an hour of peace per day, and that even a chaotic mind is still capable of creating something pretty. So that was really the main thing for me. That's why I continued doing this at the time. I didn't really care if what I was doing is right or not, the technique or whatsoever. I just painted all my anxieties away there. So every day I created more and more of these when I had time. And I started doing, more detailed paintings. And after 6 months, I was able to paint this one. So I wanted to create something with a Christmas theme. So I made this composition, and I found this really funny because when I looked at it, I saw that these are the specimens that I was showing my students, the bracts, the modified leaves, and the pine cones. So here again, I didn't use any reference photos. So these are just from my mind. But I know that I was drawing inspiration from probably one or two of my students' drawings back when I was teaching Bio 101. So there, and I found it was so hard pala to draw these things that we're asking the students to do, like painting these pine cones, for example, took a lot of time. But I was really concerned with, making it look like pine cones, because that's how I know pine cones to look like, diba? With all of the different parts and all. And I think that's really how I do it, because every painting, starts as a sketch there. And I really love seeing them come to life as I slowly paint them. And I paint very, very slow because, like I said, this is my first time doing it. But what I'm really proud of is that once I start a painting, I finish it no matter what. Even though I feel like it's not really going well, it might look ugly in the end, or the composition is wrong. Whenever I start something, I make sure I finish it, no matter how long it took there. And during this time, I wanted to see how I progress. So I placed my very first painting, and my painting, around 8 months later. And I realized, oh my God, all of the people who have been commenting all of these nice things on Facebook, maybe they're just lying to me, or they were being just really, really nice, because, oh my God, is this really something that, people would buy, like what they said? So that was my thing. I was having a lot of self-doubt because nobody who knows art had seen any of my paintings. So these are all just my relatives and friends commenting there. So during this time, even though people were asking if they could buy or if they could commission a certain artwork, I wasn't confident enough, because I didn't know if what I was doing was actually good or not. And that all changed when I was introduced to Mambing Famoso by no other than A.D. Lilian. So Dr. Rodriguez connected me to Mambing Famoso, who is the founder of the Philippine Botanical Art Society and the Philippine Fauna Art Society. So we got to chat on Facebook, and she was asking me all of these things about my paintings. Are you doing realism? Is it impressionism? And I was like, oh my God, I don't know any of these terms. So I just showed her my paintings. And, she really had a lot of nice things to say. She said, I couldn't believe you just started. These are so good. Maybe you can teach me how to paint using watercolors, because her medium is acrylic. So imagine, it's the founder of all of these art societies asking me if I could teach her how to paint using watercolor. And I really feel, somewhat, I felt validated after I spoke with Mambing there. And some of you might remember her if you're from IB, because she had an exhibit also in the College of Science, where she showed all of her paintings of the native plants and native animals. And I got to pose with her painting of Rafflesia. And this is her painting of the Medinilla flower. So there. So after that, after I talked with Mambing, I think I had the confidence to, accept now commissions, or even sell my paintings there. So this is one of the commission pieces. Many people really like this because, I don't know, maybe it's because of the composition, the color, but that's one of the first commissions that I had. And then this one, I'm really proud of this one because this is the first time that I painted something other than flowers or leaves. Yeah, so eto siya. The Cebu flower pecker, which is an endemic bird, which is also critically endangered. So this one. And I spent so much time on this because I am really so concerned with the details, like if there is hair, I wanted them to look like hair. If there's wood here, there should be lenticels that I'm seeing over here. So I'm really concerned with the details. But to me, it's not because I wanted to achieve, realism. It's just that I wanted to be believable. So it's all about believability for me. So if I can make, people believe that these are hairs, these are, lenticels on wood, this is a fruit, then that's it for me. I'm fine with that. And the problem is when people wanted to buy this painting, I don't know how to put a price on it because I really don't know how you put a price on your time and your effort. Because even though it's just one petal, it really takes a lot of time to do it, and all of these things. So that's my main problem with this painting thing. I don't know how to put a price on my artwork because even though they're not that big, because they're very detailed, it takes a lot of time to create even just one small bit of it. There. And this is my other painting now with the anahaw, medinilla, and a sunbird. So this was from an Instagram collaboration with 12 other Filipina watercolorists there. And then after, it's been what, almost a year after I started painting, I was able to join online botanical art exhibition where this painting of mine was accepted. Again, it's medinilla, a native plant. So I really love the flowers of this plant species. That's why I have painted it twice already. There. And then I also, I go back and forth between the botanical illustration part and the really loose style of painting wherein there's more flexibility when it comes to the things that I put in here. So here I just use my imagination, all of the things that I want to put in there, imaginary flowers to flowers that I know I recognize from somewhere. So these are some of my loose florals, the work. And I really love doing this. It's very relaxing for me because, it's just, imagining all of these different shapes of the leaves, all of these maybe flowers that, hinuhugot ko lang from the air. But I also think that because of the things that I know in plant morphology, it's very easy for me to think about all of the different shapes of the leaves that I'm going to put in. So I go back to this one of what we teach our students about the different shape of the leaves, the different shapes of the margin there. So I use this when I paint and didn't really notice it at first, but I'm really drawing inspiration from the things that I've been teaching my students all of these years. So when I make certain paintings like this one, wherein I needed to fill in a silhouette, it's not hard for me because I can think of, 100 different ways to paint leaves, for example. There. So this is another commissioned piece. So this is based from the actual photo of a mother and daughter, which she asked me to use the silhouette to make this one. And her only request is I use colors from this palette. There. So even though the details are very small, I really take the time, to paint even the smallest details. That's why these took a very long time to make. There. So this is my process. So first I study the morphology of a particular flower, for example, the leaves and the roots, and then I make a sketch. And then I plan the colors that I'm going to use there. And then afterwards, I start painting there. So I really love, seeing, just a certain sketch again, putting in all of the colors and then seeing it come to life in my eyes. So dahan-dahan na process siya. But of course, because I've been teaching plant morphology for more than a decade now, I'm really super concerned about all of the different structures. So for example, I know that orchids would have these types of roots. So I make sure that it's reflected in what I paint. If they have these types of leaves, then yeah, it has to be in my sketch, in my painting. And I've never taken so much time painting bark for this particular commission piece. And sometimes, I like seeing it in black and white as well, because it reminds me of the kinds of things that my students draw in our class, because they only use your pencil and paper. So there. And yeah, so I really just like painting all of these things, and at the same time, because they look so pretty. Maybe that's why I like painting flowers. I really like the pretty things there. And question that I always get, why haven't I painted Rafflesia yet? This is the flower that I study. Why haven't I painted it yet? Well, it's because I'm super intimidated by this plant. I'm so scared that my painting would end up looking like donuts for some reason. Imagine, it's the plant that I study, and yet it's my worst painting, something like that. It messes my mind. So I haven't even attempted painting this plant which I study there. So my advice to those who want to try painting is to just go for it, just don't be scared to try something different. Yeah, I was in my mid 30s when I started painting. So you're never too old to try something new or to learn something new. And if you are want to try painting at this point, I suggest that you invest in a good watercolor paper. Yes, I didn't know this before the pandemic, but there's such a thing pala as watercolor paper. You just can't paint on, osdo paper or there. So invest on really good paper, because you use watercolor with water. So the paper has to be good so that it won't buckle, it won't be destroyed while you're painting. And then if you still have money, then invest on really good paint. And then the last thing that you should spend money on are the brushes. Like, you can use any. I use just 30 pesos brush sometimes, and it can make it work. So it's paper, paint, and then brushes there. And also, if you want to paint, want to really go into botanical art, I suggest that you use the actual plant specimen instead of using photos, because I feel that the colors are better if you see them in person. Sometimes the colors that we find in nature, they're not really reflected well in the photographs. So I suggest you just use the real specimen. So you find a way to preserve it, because they will very easily. So I suggest you paint them while they're potted there. So haven't thought about this. I cannot imagine myself bringing the watercolor materials to the field, for example, to paint the flowers that we find in the forest there. But that's my advice. And as for my future plans, of course, there's so many things that I want to paint, but one of the things that I want to actually make is probably a manual, because I've been teaching all of these courses for such a long time, and we really don't have a manual, manuals with all of these illustrations that would be helpful to the students. So maybe this is one of the things that I can do, but not in the near future, because it's very hard to find time to paint nowadays, because teaching, research, being a mom and all. But probably one day this one. So that's it. Thank you for listening to my talk.
Maraming salamat po, Dr. Erika. Sobrang nakakamangha yung mga artworks niyo. Our chat rooms right now are flooded with all of these comments about how beautiful your artwork is. Thank you for such a wonderfully inspiring talk. And honestly, of course, we understand that it's a journey, it's a process, but you make it look so effortless po. So thank you. But I would like to take this opportunity to remind everyone to please visit the Institute of Biology exhibit. Dr. Erika's work is also featured there, along with Dr. Joyce from earlier, if you recall the artwork na pinakita kanina. Both Dr. Erika and Dr. Joyce are featured prominently at the Institute of Biology exhibit. You still have this weekend to drop by. Libre naman po siya sa public. So with that, thank you very much po sa lahat ng speakers natin. It was really such an amazing afternoon. We learned so much talaga, and I think we're coming away with this with a lot of thoughts, no, on exactly how art and science can and often do come together in our lives. It's very heartening also din po to know just how it also helps us, stay healthy rather. But yes, so now that we've gone through the talks, we hope kayo naman, yung audience po natin, ay buhay na buhay. Uni, can we start off with our Q&A session for everyone?
Yes po. So, gaya nga po ng sinabi ni Sir TJ, we hope that you guys are still alive and are still kicking because it is now time for our Q&A session. So, we will have, I think, 30 minutes-ish for our question and answer portion. So, if you guys have questions for our speakers about their earlier discussions, please feel free to send them sa ating chat box or via direct message sa akin tsaka kay Sir TJ.
Oo, pwede rin.
Ayun po. May nakita pa akong question kanina from G. Tinanong po niya kung kay Dr. Erika po. She said that the artworks are so beautiful. And if you have any favorite watercolor paints or brushes or paper po.
Okay. Right. So for watercolor paints, yeah, the best ones really are the Winsor and Newton ones. They're the most expensive ones, but they're really the best ones. I'm so sorry. They cost a lot, but the pigments there are really very, very good. You don't need a lot, so you can invest on even a small tube and it will last you for a very long time. For the paper, it's really just, the French paper is Arche. It's the best. That's it. It's the most expensive ones that are the best. I'm so sorry. Wala talagang substitute for those two.
Okay po. Thank you so much po, Dr. Erika. And I believe si Dr. Eusebio po ay may tanong po for Doc Yas regarding what plant kaya could be the possible source for the Viva Magenta, the 2023 Pantone Color of the Year.
Well, actually, yung isang endophyte na in-isolate namin from the rainbow eucalyptus produces a magenta colored pigment. And it's the one that it produces in abundance. So we have a few vials of it. So yun yung pinakamadami namin. So I guess the answer there is your rainbow eucalyptus. I mean the fungi from rainbow eucalyptus.
Thank you so much po, Doc Yas. And may nagtataas po ng kamay. Yes po, Miss Red.
Hello po. I would like to address this question po kay Prof. Yas po. Since yung sa study po na pinashare ko kanina ay mas yung endemic plants po for to make yung dye to dye po yung clothes po ng Filipinos. Kung may alam po ba kayong study na kung bakit yun po yung specific plants po na ginamit nila, kung may cultural or historical parang context po ba na yun yung colors na nilagay po nila. Ay, oh my gosh, sorry, yung colors po na ginamit nila. Tapos, kasi since yung topic po natin ngayon is parang a marriage of science and art, parang maganda din po na ipasok yung parang aspect po ng anthropology na dun po sa study po ninyo.
So basically yung unang source namin nung indigo is from a community. They're called the Namarabar community. And ang business no one of the source of income no community is actually to make natural dyes. So you're right in terms of yung anthropological aspect nung dyes, because it's a tradition of some of our indigenous community. We just focused on indigo because it's one of their bigger products, and then also it's also one of the priority dyes of PTRI, because we wanted to, in a way, study the work of the community as well as, of course, put science into it. So we wanted to relate it with the work done by PTRI. There are also other sources of natural dyes that are being used by the community in order to produce yung mga textiles, yung mga thread that they use to in their weaving. So marami yun. But for now, again, like I said, we were focusing on the indigo because it's in terms of economics. No, this is where the products will come from. But in terms of our future work with PTRI, we will be doing whatever we accomplish with indigo, we will be applying it to other natural dyes, natural sources. Talisay is one of the natural sources of our dyes. There's also another plant. It's like a yellow flower. I forgot the name. It's also from Abra. So we have producers from Abra, from Bukidnon, and also from, I'm forgetting this one place, and we're profiling all of them. But the point is, they all have, they're rooted in traditional use of the indigenous community.
Thank you po.
Ayun, maraming salamat. We have a question na pwede rin sagutin po ni either ni Dr. Joyce or Dr. Erika. Nasa Zoom po. From Ira, is it possible to paint the flower using the dyes from the actual flower as paint? Yes, ma'am. Either of you po, pwede pong sumagot po.
Yeah, I think that's absolutely possible, but kailangan yung paint, yung color nung nakuha sa kanya ay parehas din nung color nung let's say flower nung halaman na yun. Otherwise, hindi na uubra. Parang iba na yung magiging color niya. So, yeah, pwede naman.
I guess it's more of a chemistry question as well. I think if you use if the dye coming from that pigment in the flower would actually stick to the medium that you're going to use. And so I think not all pigment will stick to a paper or to a textile. So I think there is a chemistry behind it. But that's a great idea, actually. Why not paint the pigment of a plant or to paint the plant? So I think it's a great idea. And I think it's something that can be investigated scientifically and develop innovatively. And yeah, I think that's a great idea. Thank you for that question.
Thank you so much, po. I actually agree with Joyce in terms of, it's being a very good idea. It's going to be a really nice exhibit, actually, that you're painting a flower, and then if you use a pigment from the plant and it doesn't really resembles the color because of, let's say, oxidation through air or some chemical transformation, it's actually like a very good study piece. You're presenting a flower but a different color that was generated by the plant itself, but because of chemistry, it changes the color. It might even be color changing. You don't know. Right? Upon further exposure to the environment, the color might change. So it's a color changing painting. So that's a very, very, I think, nice concept for an art exhibit.
It's an immortalizing of flowers.
Right, exactly.
Yeah. So there's a lot of technical challenges with that, and I think it's a really nice thing to do.
Tsaka marami din kasing pwedeng i-test siya sa maraming klase ng pwedeng canvas, pwedeng paper, pwedeng fabric. So, makikita rin talaga kung saan siya magandang gamitin. Ayun.
So,
maraming test na pwedeng gawin.
Yes. Titingnan yung mga
Thank you very much po. We have a question naman po from our Facebook live stream from Voltaire Veneracion. Where can artists and creators learn about the latest researches of the UP College of Science? I think we can answer that for ourselves, but please feel free to join in. You can make a beeline for the science communications team ng UP Diliman College of Science. We are available at media at science media at science at our email, and we'll send our email through the no, but yes, and it's on their website. So we have a science communications team that can help interface the public at large together with people who are interested to find out all the many things that are happening on the campus. Pero sorry for that small promotion. Our speakers, baka meron din po kayong mga ibang mga ongoing or tapos ng works that you would like to promote. Pwede po nating ibigay po sa kanila.
I always like promoting the works of my students, though, but I'm not sure, maybe not related to art, but we've been studying honey from forest sources. So that's something that we would like to promote because we would like to say that our forest honey are competitive maybe to the Manuka honey. So, and we see that it has a lot of metabolites that are interesting, and maybe when we're done with the publication, we can let the science communication group publicize our work because it's really interesting.
Sa amin naman, the IRL Diliman, we have copyrighted materials of science designs. So we have a project called Real Data Arts. Is a way of using science to communicate things like chlamydia. There's a project like chlamydia is not a flower. We want to communicate to the public the perils of chlamydia infection to public health. We also have gut feeling. This is our studies on gut-brain axis. So yeah, I guess that's something that we want to pursue. Real Data Arts project. Thank you.
Salamat po.
We also received a question po for Dr. Ben. So for Dr. Ben po, in this generation, is there a bright future for artists and scientists in this country? And are these professions being prioritized in today's world?
For artists and who want to do art, okay, or scientists who want to do art. Actually, maraming mga there's a lot of forums for in which in which this could be done, no. Kailangan lang ng mas malaking yung parang promotion on especially within the academic community and beyond, no. So let's say if you're an artist, then you need to promote your art, no. If you're in the visual arts, then probably it takes, di ba, medyo mahal kasi magkaroon ng mag-organize ng iyong show, di ba, one-man show. You may have to get sponsors. For us who are writing, similar challenges also, di ba? For example, I can plug my book. Maybe I can share it now. Pwede ba?
Oh, yes, sir. I think that's a great opportunity to promote your book.
Okay. My book is published by UP Press, and it's sold through Lazada and Shopee, no. One way of making sure that more people will be reached out is in the case of writers, no. They get to have their works published, no. So my book, Between the Coral Tides, actually was much inspired by Steinbeck and Ricketts, which I talked about earlier, no? Discussed about earlier. Yeah, of course we need publishers, no, who will publish our work kasi we can keep on writing and writing, di ba? Tapos it doesn't get published, no? Now, limited yon, no, who's going to read it, no? Especially for writers, it's important that your craft is read or your art is read by people, no? By readers. Now, if you are in the visual arts and performing arts, then it's important that your work is seen or viewed. So hopefully, maganda sana kung may mga exhibits. So actually, the College of Science started to have exhibitions of the works of its scientists, no. And I hope this will continue, no, beyond the College of Science. Perhaps you can get the DOST and the NCCA on board to feature the works of scientists who are also doing art, or the other way around, okay? Artists doing science. Ayun. Sorry for the shameless product. We need to support UP Press also. Yeah. Kasi UP Press is the just to say what UP Press is all about, no? Yung mga ginagawa or things that are made by our faculty and staff of the university and students, the UP Press will publish it, no? And if we buy the books from UP Press, then we can expect more students and more of the faculty to have their works published in UP Press.
Sige, thank you very much po. May discount pa rin po ba sa UP Press kapag birth month? Tanong ni Michelle.
Tanong lang natin sa kanila. Before the pandemic, meron eh.
Sana. I see Red raising her hand again. Yes, please, Red.
Hello for Doc Ben po. Ask ko lang po how, since pag marine conservation or marine ecology po yung usapan, paano yung pag-promote po ng advocacy for marine conservation, mas po sa photos, kasi I have friends po na underwater photographers po, pero nakita ko po, first time ko po makita yung yung book niyo po at saka yung book ni Steinbeck po na writing about marine conservation and marine ecology. Paano nyo naman po inyong mapapromote po among marine scientists or marine conservationists po yung pagsusulat po? Kasi di hamak po mas madali kumuha ng photo kahit na mahal kasi yung equipment, yung pag-dial, ganyan. Pero di hamak na mas madali kung mayaman ka na mag-picture kesa po magsulat. Pero napakaganda po ng mga passages po na pinakita nyo. Iba po kasi dating kapag sulat po yung pag-explain po.
Kasi iba naman ang literature is a different medium, di ba, compared to the visual arts, no. Ngayon, ayun sabi mo, marami nagpi-pictures. Okay, ako wala talaga akong talent sa photographic composition, ha. Naamin ko na yan. So what I do, yung sa aking sinulat nga na natural history guide, I got all my friends na may advocacies in marine biology for them to agree to use their pictures for my book. So maybe that is one way of doing so. Kasi sa totoo lang, kung magpo-produce ka ng coffee table book na magagandang pictures, okay yan, di ba? Actually, mahal mag-produce ng coffee table book, no? Medyo limited din ang iyong readership kung coffee table book kasi mahal, di ba? Magkano ang coffee table book ngayon, no? So totoo lang, when I was discussing the possibility that we can come up with coffee table books in marine biology, no. So I talked to people who are book designers, di ba? Remember, it's also visual art, no. Book designers, and sabi nila, nako, kung gagawa ka ng ganyan, maganda pictures, dapat maganda yung papel, maganda lahat, maganda ang book design. And that would cost you probably 2 million. And that was before the pandemic, no? So, malaki rin yung investment, no? Sa ganyan, no? Paganda sana. Of course, you can have a book that has mas hindi masyado maganda yung papel tsaka yung resolution ng pictures. It's cheaper, but of course, it's not as attractive as So, yun nga, kung maggagawa tayo ng ganyan, kailangan ng support, no? Maybe. I think ang UP may mga support sa creative work, no? But of course, it's not enough, no? You may have to get some donors, no, to sponsor it. And I know some people who have done so. Pero not in marine biology, in plant conservation. Tsaka yung mga bird watchers, no? They have sponsors to publish their bird books, no? And one of my friends contributed about seabirds in my book, eh. Kaya nandun yung they get sponsors and raise funds for that, no? Because it costs some money to do it. And of course, hindi ka naman mag-publish ng libro kung hindi tutubo yung publisher, no? So we have to do a very good marketing campaign, okay, to have these books sold. Okay? So cooperative effort siya. Kailangan lang talaga ng kailangan natin ng kailangan talaga you have the drive to finish it, eh. Kasi you can write and write and write, it will end up as a draft. And if you don't get it published, then nobody will read it. Okay? So it's a motivation, drive. But it could be done. Marami nang gumawa, eh. Sa totoo lang. And some of them are privately published. Those who were able to get donors. Yun.
Thank you so much po, Doc Ben. And we have another question that I think any one of you can answer po. So, how can the arts and sciences work together to address environmental issues in the Philippines? And feel free lang po, and anyone po sa inyo.
From the perspective nung study namin with natural pigments, the reason why we're actually exploring natural pigments from either plants or microorganisms is so that we can veer away from the more synthetic dyes that are actually causing pollution. So one of the big thing now in terms of the natural dyes actually is really the use of indigo dyes from natural resources, kasi one of the biggest pollutant is the production of jeans. Yung mga denim jeans natin na blue jeans, right? It's actually made from synthetic indigo, yung blue jeans. So it causes a lot of pollution because it causes a lot of it uses a lot of harmful chemicals to produce the jeans. So now there's a move towards producing indigo on a larger scale, but from natural sources. So from that, I think we are doing this, sourcing out our natural pigments from plants, and at the same time trying to work in terms of minimizing the pollution that are caused by this by the industry, as well as natural dyes are also used for paintings, artworks, right, yarns and all. Marami siyang use. So I guess we try to have a sustainable sources as well as earth-friendly sources of natural dyes. So yeah.
Yeah, ako naman share ko. Actually, just last week, galing kasi ako sa Bicol. Doon, marami doong pineapple plantation. And I bought yung pineapple fabric. And one of the things that I discovered, kasi usually embroidery yung ginagawa sa pineapple na fabric. But then in Bicol, wala masyadong marami nag-embroider doon. So I was talking to the DTI representatives and I was thinking that dapat may certain independence yung Daet Camarines Norte, for example, for making the pineapple fabrics. And I was thinking of actually collaborating with Hiyas. Ang dami naming mga points of collaboration of using natural dyes to actually paint on pineapple fibers. Kasi major pollutant din yung mga textiles, and with fashion parang nagpapalit-palit lagi. So we want a more sustainable and environmental friendly medium like plant-based textiles, I think. And science can help with that, and arts as well. Thank you.
Yung yung tanong na yun, paano natin dadala sa mga tao yung ano, yung arts and yung science and art, no. Actually, 3 before the pandemic, few years before, no, we wanted to do that. At minsan yung mga foreign development agencies, sometimes they have calls for that. So titingnan din natin, no. I think TJ and I tried to apply. We were shortlisted but we didn't get the money. Ang plano namin is actually to come up with a natural history guide na maiintindihan ng mga coastal yung mga bata sa coastal communities natin, no? Kasi alam ko from talking to teachers, hindi nila although nasa coastal community sila, hindi nila alam yung marine biodiversity because of a lot of factors. Una, economic, no? They have to help, kailangan tulungan nila yung pamilya nila para kumita. And syempre, wala na silang time para maging curious and explore the environment around them, no? So what we wanted to do is put it in a sort of yung in a way na pwedeng i-introduce sa basic education in elementary schools in our poorer coastal communities, mainly in the Visayas and in Mindanao. We proposed it, we got shortlisted, but of course we didn't get to the final list. So those are some of the things that we can do, especially for writing, no. Gusto nga namin sana magsulat yung mga bata, nakikita nila dun sa sa kanilang marine biodiversity. Kasi yung perception ng mga nag-aral ng marine biology iba dun sa perception ng isang bata na nasa isang coastal barangay. Iba, maganda sana kung maisusulat nila yon, no? Yun ang gusto namin mangyari. Ayun. Kaya lang, hindi kami pinalad na kami nanalo ng grant. Maybe we can try again pag pag may call again for something like that. Ayun, Ayun.
Ako naman po, in terms siguro of yun nga, biodiversity conservation naman. So, even though, roses and tulips and cherry blossoms are really pretty, mas marami pang tayong mga endemic or native na plant species na may mas magaganda pa. So, kung sila yung magiging parang focus na as subject sa mga paintings, makakatulong din siya. Kasi from my experience, minsan yung among my friends, my relatives, hindi pala nila alam na may ganito palang ibon or may ganito palang flower and na dito lang sila nakikita sa Philippines. So I think that's also one way, parang yung information para sa lahat na meron tayo nito and yung iba sa kanila critically endangered. So yeah, we have to do our part.
Thank you so much po for answering that question. And we also received another question po for Dr. Joyce. So it is mentioned that innovation will be talked about in terms of science. So can the person ask how important the arts are in pushing for innovation?
Yeah, thank you for that for that question kasi medyo hindi ko nga yata na-elaborate kanina. So with innovation, actually we're pushing for science to actually solve real world problems, yung mga actually what the community or what people need. But so when we do innovation, we always we create a value proposition for our discoveries in sciences. But what I realize is that without the involvement of the liberal arts, where you actually think of what you do have to do customer discovery, what the customer will buy, you have to market an idea and a product. And so you would need arts for that because it's not enough that you have a technical solution. It's hard to change behavior when you're not able to empathize with who are going to use these products or these ideas. So the interaction of arts and innovation is you can have a better marketing strategy for your innovation, and then you can make more impact. So it's always, at the end of the day, you have to change behavior. When you have a technical solution, you have to change behavior. And the technical solution, presenting a technical solution in a very academic way, it's hard to change the behavior of people. So kaya kailangan talaga yung liberal arts din with that, because it will touch on the joys and pains and human experience talaga. That's my thought on that. I hope that helps answer your question.
Maraming salamat po. Nakakatuwa po no na we're looking at our audience right now and there are lots of students but there are also a lot of educators. So may tanong po tayo dito from the audience. What activities can we ask students to do for art and biology? Yun, pwede po itong sagutin na Dr. Joyce, Dr. Erika. Pero actually anyone, if you have ideas, maganda pong, it would be great to share these and hear from you.
Ano yung tanong? How to ano?
How can we ask, what activities can we ask students to do for art and biology? So I
Okay.
guess they're asking yung ano bang pwedeng interplay ng art and biology.
Well, kailangan ibalik yung training mag-drawing. Kasi ngayon kasi napansin ko na pag nagtuturo ng biodiversity, kinukuna ng pictures sa cellphone. Okay. Now, my PhD advice supervisor was completely traditional. Lahat ng mga pinag-aralan kong shells for my PhD, pinadrawing, no? Although noon may digital camera na. One reason for, kasi di ba pag nag-drawing ka, kanina nga, di ba pinakita yung mga drawing ng mga, plants made by students, di ba? Kailangan talagang ibalik yung skill na yon kasi skill of observation yon sa lahat ng biological sciences kailangan mag-observe, di ba? Pag nagkumuha kayo ng picture using your phone, hindi yun complete observation. Actually, nag-observe yung phone. Okay, tapos tiningnan nyo na anong lumabas, no? So wala yung yun nga sabi nga ni Steinbeck and the classical marine biologist, no? You really have to observe, then you have to draw. Marami yung kanilang zoological catalog, drawing, may pictures din pero karamihan drawing. Tapos susulat ka ng kung ano yung interpretation mo ng drawing based on what is scientific, ayun. So, siguro kaya ibalik yun sa mga estudyante. Huwag na sila mainis kung magdo-drawing, no? In the College of Fisheries, we were expected to draw all the fish. Okay? Ang dami nun. That's part of the classical training in zoology pa rin to draw your specimens, no? Sometimes nakakairita. But looking back, it was an important skill to develop for me to observe marine biodiversity. And I think all the botany people will also agree kasi kailangan nyo mag-drawing. Ayun.
Thank you. So i-share ko yung activity sa cell biology saka sa immunology. Yung sa cell biology, I actually asked the students, so I teach cell signaling pathway. So we asked the students to draw an illustration of the cell signaling pathway from the signal transduction to cell response. And I asked them, what's the picture of the cell signaling pathway in a disease state? For example, there is kasi yung mga diseases is a deregulation of cell signaling pathway. So they will draw that. So conceptually, naintindihan nila. So that's one activity. Sa immunology naman, we have an exercise where I will draw a skin sa board. And then it's a group exercise. All the students will go there and sasabihin ko, there is an infection, there is a viral infection. I will put a virus dun sa skin. And then I said, everything that you know about the immunological process that will happen after a viral infection, you draw this. So it's a whole whiteboard and the whole group of students will go there and draw the innate immune response, the adaptive immune response. And at the end of the exercise, we have a huge painting na whiteboard ng immunological process sa immunology. So yun yung that's the exercise I can share na ginagawa sa Institute of Biology that merges and teaches science and arts sa students. I hope that answers your question. Thank you.
Thank you so much. So we have another question, but before that, we would like to remind you that we can only entertain 1 to 2 more questions before we close the Q&A session. We've been receiving a lot of questions so far, and thank you so much for forwarding them to us. So ito po yung isang question that we saw here in Zoom. So this is from Miss Natalie Bascos. Thank you so much daw po for the very inspiring presentations. One of the things I picked up from this webinar is how informed creativity can happen in a scientist who does art as well. Can you comment on how this intersection can be encouraged in students?
I think the best way for students to be encouraged is to experience the science, right? So when they do the experiment, they observed it firsthand and they discovered it firsthand. So that's that experience, I think, is the one that's going to reinforce this intersection. So for example, yung research namin sa national dice, these are results done by students. So they're the ones who actually discovered it. They're the ones who culture the fungi. They're the ones who scale up the production of the pigment of the fungi. So the discovery is for them, and the result of their discovery is also for them. So I think experience is really the best teacher. So yung mga suggestions ni Sir Ben regarding, students need to learn how to draw, I think that's also a good learning for students, because it's experience that's going to teach them a lot. So that's for me, experiencing science that will encourage the intersection with how art is going to be used and how art is going to inspire.
Well, for literature and writing, magsulat lang sila ng magsulat. Huwag sila matatakot. Okay? To express what they see and what they feel about nature. Yun, magsulat lang ng magsulat, no? Of course, we will find, syempre papabasa mo yun and you can explain. Sabi ko sa kanila, wag muna kayo mag-isip ng writing workshop kung saan savagely criticize yung work ninyo. Basta magsulat lang kayo ng magsulat. No? That's for the literary arts, no? Siguro for the visual arts, you just draw and draw. Okay? Ayun, no. Sa STS kasi, that's what I tell students. You have to write or whatever creative in order to relate to the concepts that we teach in STS. Okay, just do it, no, and present it in class. Okay, and see what other people will say, and if they can connect with you, then that's good, no.
Thank you so much po. And we have a question po for Doc Ben. So they appreciate for the prompt of what does the starfish know. It would be interesting if somebody were to write, what do the different inhabitants of a tide pool tell each other when the moon is full?
Well, kasi I've been studying, that's my PhD talaga, intertidal ecology, and I've been doing it. May mga, I think I have 3 master students who've done it na yung, what do they feel? If you read Steinbeck and Ricketts and even Rachel Carson, yung mga marine biologists that were writing at the early part of the 20th century, all of these observations is all about sex, di ba? I keep on telling my STS students, everything that you see in marine biodiversity has some sexual connotation. Kasi sabi ko sa kanila, yung mga ano sa inyo, mga prude, I think you just have to throw it out of the window, no? Example, one of the invasive species of sea squirt or ascidian na pinag-aaralan namin ngayon, part of my research program, if Falusia. Okay, di sinabi ko doon sa mga RA, ba't yung tinawag na Falusia? Because it looks like the human penis. Tapos sabi ko, hawakan nyo kaya, tingnan nyo anong mangyayari. It actually acts like a human penis. So, how come it functions like a human penis, di ba? Sea squirt, it squirts. Anyway, sabi ko sa kanila, imagine, no, the first marine naturalists, they saw that, they immediately knew what it is and named it appropriately. So sabi ko, yeah, so how do they, everything is about, if we're Darwinian about it, everything is about living, no, as Steinbeck correctly said, everything, the whole purpose of life, whether tayo or the marine organisms, is actually for living, no? And part of that whole function is about sex. So a lot of the things that Steinbeck wrote, whether in his other fiction or in the Lag of the Sea of Cortez has something to do with sex. Kaya ano siya, it's liberating in one sense kung galing ka sa isang society na medyo hindi tayo masyadong nagdi-discuss tungkol dun, no? Liberating siya. And it gives us a new insight on biology itself. Okay, ayun.
Ayan, thank you so much po. And last question po is, ang question po is, ever since high school daw po siya, he is very passionate with literary and visual arts. He wishes to create a children's book about marine conservation in the near future. However, he's torn if he should pursue his master's because it is something that is expected from him. And also, he'd like to pursue to deepen his understanding and expand his horizon as a graduate of BSMB. But he also finds teaching and sharing information through lectures and speakership opportunities as an avenue for amplifying marine conservation. Right now, he is teaching biology, chemistry, and research, and he is able to share marine conservation with his students. What motivation can he draw from you as scientists?
I think since it's a last question, maganda po na ibuksan na po natin sa lahat ng disciplines po natin para sa lahat po ng speakers natin yung tanong na yon. Kasi maganda rin naman po na marinig po natin yung sagot. Yung ano yung motivations na pwede nating maidala, what motivations can we bring to the table based on our experiences, based on your based on your backgrounds that can help motivate students in this way.
Oh, ayun. Ayun sa tanong, kailangan niya matapos si master's degree niya in some discipline, tapos he wants to do this and that. Actually, just follow your passion. Di ba? Eventually, mag-align din naman. Actually, I found out later on when I was doing my PhD, I started writing all of these things. Actually, my book that you saw, which I shamelessly plugged, actually is, for me, that's the most important publication from my PhD. It's not all those cited publications na who's gonna read it anyway. Ito talaga, it took me about 20 years to have, kasi I finished my PhD in 2023. In 2003. And the book came out in 2023. 2022 pala. So it's almost 19 years, no? So just because it was my passion, so I just followed it, no. Ang ginawa ko is to take out the academies, as they say, and put it in a more introspective and reflective way. And the book came out eventually. So ayun, just follow your passion, and it would lead you into a convergence. Siguro makuha mo yung master's degree mo, and you'll be able to do what really means to you. Ayun.
Yeah, I guess what I can share is yung sa journey of doing science, if that's what you want to do as well, and arts. Siguro one at a time. I think focus on something that you become a really expert in, especially kung gusto mo maging scientist. I think yung discipline talaga ng PhD is required to do science. And maraming time ang kailangan for that, and you have to focus one at a time. And then when you want to do arts, it will follow. I mean, if you're naturally inclined, kasi what happened sa akin, hindi naman matagal yung training ko sa arts. I have some fundamentals from my high school teachers, and magaling yung high school teacher ko sa arts, the late, yung teacher ko sa art. So I think isa-isa, and focus one thing at a time and do something. If you want something so badly, just work hard on it, and you can achieve what you want to achieve. There's no substitute for studying and working hard on something that you want to be. So I guess that's my take on that. I hope that helps. Thank you.
Okay, I think that's the hardest question of all. Because each one of us have different paths in life, right? And the path that one takes where one will be successful is going to be different for another person, right? The path that we took where we are now or where we have been might have been the path that was suitable for us at that time or yung current life namin ngayon. And it's going to be different for everyone. So ako, I believe in destiny. Medyo ano siya, for a scientist, that's a little bit weird, I guess. But I believe that there are certain duties that you are supposed to fulfill and those will come to you naturally. Hindi siya pipilitin. That you will realize it at the right time when you are ready to realize it. So to me, I believe in that there are certain duties that we have to fulfill or there are certain dreams that we can fulfill at the right moment in our life, at the perfect time in our life. And nobody can tell you when that time is. Ikaw lang at tanging ikaw lang. So whatever you decide, the best decision will be made by you and not by, copying other people's decisions. So yun. So that's why I think this is the hardest question ever.
Naman, I agree with Mom Joyce. Like, it's really good for someone to excel at something, to be an expert at something. But I think it's also really important to be multi-passionate. Like, if you have more than one interest, then really just go for it. Yun lang po.
Ayun, maraming salamat po sa inyong lahat. Huling tanong na nga lang, napasabak po tayo. But it's really, sobrang nakakatuwa po talaga, nakakataba po ng puso na napakarami pong tayong engaged na listeners and viewers today, this afternoon. Pero syempre, sabi nga po nila, all good things and all that must come to an end. So we're wrapping up. To those who joined the discussion and asked their questions, thank you very much. Dun sa mga hindi po namin nasagot, okay lang kasi we will be sharing an evaluation form later na pwede niyong gamitin to send your questions to our resource speakers. And also, as I mentioned, if you have any other things that any other questions that na pwede naming tulungan kaming SciComm team, you can reach us through our email. We posted it already in the chat discussion. So with that, I'll pass the baton to Uni so that we can start tying up the afternoon.
And thank you so much po, Sir TJ. And as we end this very, very, very insightful webinar, I would like to call on the Assistant Professor, Dr. Michelle Eusebio. She is an STS1 professor, an archaeological chemist and anthropological archaeologist at the UP Diliman College of Science. And she earned her PhD in anthropology, majoring in archaeology at the Department of Anthropology, University of Florida in 2016. She also earned her MS Archaeology and BS Chemistry degrees from the University of the Philippines at the Archaeological Studies Program, now School of Archaeology in 2010, and Institute of Chemistry in 2002, respectively. So I would like to call on Dr. Michelle Eusebio.
Okay, good afternoon. Thank you very much for coming, despite today being an impromptu special non-working holiday. We congratulate and thank the organizers of this event in conjunction with the celebration of the National Arts Month. We are very grateful for our resource speakers for sharing their expertise and experiences in integrating science and art. They have proven that science and technology are not separate from art and culture, this separation being reinforced by media and sometimes everyday life. A careful look at history will show that science, technology, art, and culture are always intertwined. The important aspects of science and technology are actually rooted in cultural perceptions of the world and of the different ways through which its material content, environment, and people are understood. So, napakaswerte po natin ngayon kasi yung mga kumukuha ng STS1, meron ng isang module on the cultural aspects of science and technology that includes yung topic ng webinar natin ngayon, yung intersection of science and the arts. Kasi nung nung mga more than 20 years ago, walang ganyang module sa STS. So parang mas exciting ngayon mag-STS. And for sure, a lot of our STS1 students are here with us kahit holiday. Ayan, at walang pasok today. So yun, nakita nga natin yung mga ilang examples from our resource speakers. So kay Professor Ben Vallejo, yung intersection between science and literature, and probably ma-realize natin na at may mga nakita na tayong examples na writing non-technical stuff actually helps writing yung mga requirements sa aming faculty members sa mga publications na yan. And there are actually some scientists na matataas yung H-index or mataas yung kanilang positions na. That on the side, they're also actually writing novels, literature, nagko-compose din ng music, and some of them are bloggers pa. So, and nakakapagtaka talaga na with all of this writing sa mga journal publications and books, nakakapagsulat pa sila ng academic side and perhaps siguro scientific non-technical ano ba sasabihin yun? Yung blogging or writing, sana ma-encourage din, and siguro naman, sana may merit points din po yan, di ba? Okay, and yung kay Dr. Joyce Ibana, so, and nakita natin yung intersection ng microbiology tsaka scientific illustration, at tsaka yung ano din, nagagamit natin yung art through painting for science communication. And siguro yung pinaka takeaway natin from Ma'am Joyce ay yung impact equals the intersection of art, science, and innovation. From the talk of Ma'am Hiyas Junio, yung chemistry behind the colors for expressing art, maganda to kasi nagkaroon tayo ng more awareness na yung mga colors na pinangkukulay natin ng kung ano-ano, like yung mga tela at iba pa, ay hindi lamang galing sa mga synthetic sources, kundi pati na rin sa natural sources. And it's really good na na-highlight sila, and she also brought up din yung sustainability din on using these natural dyes, kasi yun nga, kapag yung source nga ng mga common natural dyes ay yung mga plants, pero some of them probably are critical species na baka mag-endanger na, so maiging may alternative din, pero from nature pa rin. Okay, and lastly yung kay Ma'am Erika Bascos, so and perhaps ma-encourage tayong lahat na yun nga, mag-resort tayo sa art. We really don't have to be yung artist person na nag-exhibit sa mga galleries, but something to help us din with our mental health, kasi hindi lamang po students yung may mental health issues, pati rin po tayo mga faculty and researchers din, so kailangan natin ng diversion, kasi grabe yung pressure sa atin maliban sa pagtuturo, nandiyan din yung research pa extension. At yun nga, di ba, so ito talaga yung testament na kapag biology major ka, may minor ka in fine art. Talaga, kasi kasi ako as a chemistry major back in the day, we also have to take yung second general biology kung saan may taxonomy. Ang dami talagang drawings doon. Ako kasi wala akong talo sa drawing, halos sumuko doon. Pero yun nga, maganda talaga na maipakita din through the art of scientific illustration yung mga different species natin that comprise our Philippine biodiversity, and also then to emphasize din na hindi lahat nakaka-capture ng camera kasi ng picture. So maigi talagang may corresponding scientific illustrations din, kasi yun yung doon makikita ng ano ba talaga ang actual na observations din ng scientists. Ayan. So ayun. So and ang ganda talaga ng diversity na talks sa to. I think parang isang module na to sa STS or Science, Technology and Society. At saka yun, to end yun, mangungumpisal lang ko na actually, it's actually art that brought me to archaeology from chemistry. Kasi yung undergraduate seminar topic ko ay tungkol sa wall painting si Queen Nefertari. Ayan. Kumbaga, yung pinag-aaralan doon yung chemistry ng pigments na nasa wall paintings na yon. So, again, maraming salamat. Maraming salamat sa mga nandito, sa mga congratulations sa organizers. Yun lang po. Magandang hapon.